Hello everyone, welcome back to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. I'm Ken Hansen, Associate Professor in the Department of Chemistry and Biochemistry at Florida State University. I do all things light matter interactions, photons driving processes like electron transfer in solar cells or photocatalysis, bond breaking, bond making. But more importantly, joining me today is Dr. Lisa Chadwick. Lisa, if you wouldn't mind introducing yourself.
Hello, I'm Lisa Chadwick. I am a program director at the National Human Genome Research Institute, which is one of the parts of the NIH.
All right, so equally important, what game are we starting with?
Well, we're starting with my favorite game, Metroid.
This is your all-time favorite game?
Pretty much.
This is the go-to.
Yeah.
All right, ladies and gentlemen, this is Metroid. I think this was one of the really early releases for the NES.
Yep.
So 1987. We're gonna see if you remember the maps.
Well.
I mean, what sucked when we were younger is there was no like YouTube. There was no.
Nope.
You know, there was a Nintendo helpline that you could call and charge like $2 per minute to your parents, but.
Okay, I did not even know about that.
That's for the best.
But what I do remember doing a lot is reading long text walkthroughs on whatever web page. I mean, there was like some place where you could go to get walkthroughs.
Yeah.
And they had all sorts of like ASCII art and everything, and you had to like scroll. Yeah.
Yep.
Yeah, so I did that a lot.
Calm down, Bronco. Welcome back. Presumably you're in Massachusetts now. Pretty close to it. Grad school starts pretty soon.
Good luck in grad school.
Absolutely.
Oh, don't, jeez. I am a little concerned about my ability to speak intelligently while being extremely
no worries.
Intensely concentrating on this game. So
that's part of the fun.
This might not be my best Metroid playing ever.
Yeah.
You can't judge me on that.
One of the things I really like about this outreach is how humanizing it is.
Yeah.
Right? Like you are knocked down off your ivory tower immediately.
Right.
Direct in a video game. Like, it's a good thing we don't do multiplayer online competitions.
Oh, that'd be fun. We could have like a different like discipline, academic discipline, like let's make
like
that
Smash
happen.
Brothers or
yeah, I'm here for that.
Mario Kart's been one of the most popular games we've had.
I mean, I've definitely played more than a few rounds of Mario Kart, but that I really am a little bit too intensely interested in. And I also watched a little bit of last week.
Yeah.
When she was playing Guitar Hero, another game that I've played quite a bit. But there's no way I could do this and play Guitar Hero at the same time.
So I've had two guests doing Guitar Hero, and the first one, Christina Holmes from Chemical Engineering. She was, or Biomedical Engineering, sorry. When she suggested it, I'm like, all right, we'll give it a try. But turns out she was better than than her other games, so she did it.
Okay, someone is in the chat named Drosophila, which is a fruit fly researcher, I'm assuming.
Yeah, Drosophila related.
Yeah.
That's fun.
Good username.
Drosophila, welcome to the stream. First time chat. Thank you for being here. You've had time to settle in. So if anyone has questions for Lisa Chadwick, feel free to throw them in chat. She's an NIH program director, does funding related to genetics, genomics.
Yeah, so I can tell you about some of my sort of areas that you could ask me questions about. One is rare disease genetics. So I lead a program called GREGoR, which stands for Genomics Elucidates Genetics of Rare Disease.
It's not a smooth acronym.
I know. It's pretty good. Well, GREGoR, so I was really proud of this because GREGoR, named after Gregor Mendel.
Oh, you named it.
Yeah.
Nice.
Well, okay.
Oh, so you backronymed it.
I helped. And I actually, my institute director is the one who came up with the really, I can't get in there.
Yep.
He's the one who came up with the really good GREGoR Mendel acronym. So we mostly study Mendelian diseases, which are single gene genetic disorders. And what we're doing in GREGoR is developing strategies to speed up that process, and we can talk about that in more detail later. And then I also do work with people who are trying to get NIH funding, and I can talk all about that. I see there's a question in the chat about that.
Yep.
I actually love doing outreach on that. I do grant writing workshops. I run NHGRI's early career researcher programs. So I have had a couple of RFAs that are for researchers who are early stage investigators, which is within 10 years of getting your PhD.
Yeah, so we'll start out with that. Calm Down Bronco wants to know, as a hopefully future academic, what makes a great grant proposal, and how does the NIH decide which to fund? So maybe we should do a cradle to grave recap of like just an R01 grant.
Right. Okay. So most of the time you wake up in the morning and you're like, I've got a great idea for a grant that I want to submit, and you write it. So and it takes you.
So I'm gonna interrupt because that's a dirty lie. The answer is I have a deadline coming up. What proposal can I write to match that deadline?
That's true. But the R01 in general, well, okay, there's I don't even think I can go through this yet, but
yeah.
The R01 is like the major sort of bread and butter grant proposal, and you can submit R01 proposals through specific funding announcements that
oh no. No,
that are I know.
It's better to die.
That are asking for specific kinds of research, but otherwise you can just like write them on whatever topic you want, which is what the majority of applications I think are. And then it gets assigned to one of the. Oh, I probably have to. Oh.
Yeah, that was it.
Wow. Okay.
Do you want infinite health? Rockets. Well, we cheat all the time on this channel. There's no pride here.
Yeah. Well, I would hate to play this and that one large column.
Again and again, yeah.
50,000
That's usually
times.
why we do it. So you're going to have infinite health. You don't have to worry about dying. This is still a hard game because you got to figure out where to go.
Okay, so you write your grant proposal. What?
You got to get your roll.
Oh, yeah. No, I have it.
Oh, it started you over with it. Huh.
You write your grant proposal and you submit it, and it gets assigned to one of the NIH institutes. That depends on sort of what kind of science you're doing in the proposal. And it gets assigned to a review group. And those are your peers who are going to review your grant proposal. And we have a lot of what are called standing study sections, which are panels that meet 3 times a year. It's largely the same people on the panel every time. You serve like a number of year. I almost said sentence. I probably wouldn't call it a sentence, but
off the record, it's a sentence.
it can be really helpful to you to serve on a review panel.
Yeah.
But they review grants and give them scores, and then we program staff get the scores, and we basically most of the time fund in score order. Sometimes we fund according to certain specific scientific priorities, like if an application isn't clearly within the funding range, but it's doing something that's really important or we see as a big gap in our portfolio, that is more likely to be funded. So that's sort of the basic process. Some of the things that I think make a really good grant proposal are, number one, don't write it like you are reading it, right? You're writing for a much broader audience than your specialty. The reviewers who review your grant are not you. They don't know everything that you know. And a lot of times people get a little too in the weeds or too technical, and you lose people who have like a good working knowledge of your general area, but like are not a specialist in exactly the same thing as you are. So it needs to be understandable by everybody. And I always tell people to have your colleagues read it, and not just your closest colleagues that do very similar things, but people who are doing things that are kind of reasonably in your area, like molecular biologists, right? But not exactly the same as you, because they'll be able to tell you like, oh, I didn't understand why this was important, or why we would want to do this. The other thing that I think is really important, and I hope that everyone will do, is contact their program officers before you write a grant application. Oh, that is Gigi. A lot of people don't contact program officers, and I can give you a lot of feedback before you submit a grant proposal about whether I see like major sort of deal breaker kind of flaws in your aims. Like maybe you've written 3 specific aims in your grant, and the last 2 will go nowhere if the first one doesn't work.
Yeah, codependence.
Yeah, and people do that all the time. Or you write 3 aims, but basically what you're proposing to do is solve the entire field of genetics in one grant application, which is probably unlikely to happen in the course of 5 years with a budget of $500,000 or less. And then I can also give you feedback about what study section is most likely to review it. There are some other ways that you can figure that out, but I listen to a lot of study sections. I know kind of where this is probably going to go. And then you can look up who is on that study section, so you know almost exactly who the audience is that you're writing for.
And you can suggest which program and which study section, right?
You can put recommendations in like a cover letter that you submit. And I actually am embarrassed that I don't know a lot about how that happens, because I just see the applications when they come in. I don't know how you get them there. But I understand that there is some sort of oh good, now I have muscles. There's some sort of a cover letter where you can suggest the most appropriate institutes, and you can suggest study sections. They're not always going to listen to you. There are a lot of factors that play into what study section you get assigned to, like what their workload is, what they see as their scope versus other sort of similar study sections. But so it's helpful to sort of have an idea of a couple of different study sections that might be reasonable, and that can help you plan writing your application. Okay.
Oh, no, that sounds... NIH is interesting because they tell you who the reviewers are. Like, it's one of the few funding agencies. Well, they don't tell you the specific reviewers. They show you a list of who's on the panel, right, or who's part of the study session.
Yeah, where is your research funded by?
Mine's mostly NSF or hopefully DOE.
Okay. And
Army. Yeah.
So they don't tell you... They don't tell you anything?
No, it's entirely anonymous. They don't give us any information. And the other thing that's interesting about the NIH compared to the others, because I've applied there, it was a long shot, but I gave it a shot. But you're arguably the most objective grading system. I mean, in terms of your scoring. Good or bad?
There's a lot of people who have written grant applications that were not successful that would probably argue with you about that. But no, reviewers, like, as much as people like to complain about reviewers when their grants don't get funded, like, reviewers are people like you, they're your colleagues, and they're always trying hard to do the best job that they can. They're trying to be fair. They're trying to find the best science. They have like a day job now.
Oh, look at that. Veteran.
Oh, but I don't have bombs. I can't.
Can you jump out of the...
No, because I need bombs.
To blow up the path. That was still a veteran move. Kudos.
Thanks. I gotta remember where bombs are.
No. That's fine. You got help.
Okay, I don't remember what I was saying. Oh, but they they go to work every day. They run their labs. They write their own grants. They write their own papers. They teach. They grade papers or whatever they do, right? They come home. They feed their children. They put their children to bed. They, now it's like 10 o'clock. They have finally had 5 seconds to themselves. And what are they going to do next? They're going to sit down and read like 7 grant applications that are not short.
Yep.
So they really, this is not good.
No, it's fine.
What is happening here is not good. Okay.
This is what it's like to review grants.
I can't even get out of here. Okay. I don't know how to.
I think you can get up there.
Yeah, that's. Oh. Okay, good. Okay. Right. So that's also something that you should think about when you're writing your application. People in science are used to writing to sound as smart as they possibly can.
Dense.
And dense, right. And they write really long words and they don't want to use white space because they're like, I've got to use every piece of this paper to tell you everything I know about this topic and why this grant is so important. And the person who is reading it at 11 o'clock at night does not want to be faced with a wall of technical text. Can I jump up there?
I think you can rocket these. Can't you? You have rockets, right?
Oh yeah, but how do I use rockets again?
I honestly don't remember.
Is it select? Yeah.
I might switch to rockets. Oh. Those guys are temporary, right?
Oh, okay. Okay. All right.
Perfect.
So anyway, I hope that was helpful.
Yeah. The advice I give to junior, I'm now senior faculty after 10 years, but I don't know how that happened, but it did. The advice I give to junior faculty is the proposals are not about what you want to say, it's about what they need to hear.
Yes. Yeah, I say that all the time. You're not writing this, unless you plan to fund your own research, you're not writing it for you. You're writing it for the reviewers.
All right. Draso Killer, thank you for the subscription. Again, welcome to the stream. It's a pleasure to have you. Throw your questions in chat. You guys, you don't have to support our stream. We don't need subscribers. We do this out of the kindness of our hearts, and we like drinking and playing video games. But if you do support us, it does pay for our alcohol. Speaking of which, Lisa, what are we drinking tonight?
Well, we're drinking a rosé from the south of France called La Vendette.
La Vendette. It's rosé.
Which we got at one of my favorite local liquor stores.
Yes. Market Square Liquor.
Yep.
Shout out to them.
Exactly.
They sponsor the stream. Just kidding. They should.
Informally.
they should at this point.
This is not going well. Okay. Well.
Oh, Reese's Pieces knows this game. If you have any advice, we're happy to take it. It's been a while.
What I'm in the total wrong column, aren't I?
To get the bombs?
Yeah.
Maybe?
Yeah.
Oh man. Okay. Okay, we got time.
Okay. Can I answer that other question that I see up there?
Yes. Almost PhD done.
Congratulations.
It's optimistic. I'll give it to you. You're probably almost done. If you've had a conversation with your PI, you're almost done. Yeah, you got to go back up to the elevator, Reese's says.
Yeah.
All right. They want to know, hi, Dr. Chadwick. I'm curious to know what your typical day looks like. Okay.
So my typical day is probably 6 hours of Zooms.
And then you came here and did this.
Right. Yeah, bro. So we spend, okay, the various kinds of things that we do are, number one, talk to researchers, the ones who are smart enough to contact us ahead of time. Now, where is that elevator? I feel like I have to go. Oh. Yeah, I wish I had those. Oh, I do have the frozen missiles, probably, don't I? I think that's them. So I talk to PIs. During review season, I will be listening to your grant reviews. So after you get your summary statement back, which is sort of the report back from what happened in the review of your application, I will talk to people and sort of explain to them what my perspective is, what I think that they should do next in terms of either fixing their application and resubmitting it, or submitting a new application, or waiting and hoping that they're going to get funded. So working with grantees, that's one part of it. At my institute, we run a lot of research consortia. If you are interested in genomics, you may have heard of some of them. One of our big ones was called ENCODE. ENCODE is a really great acronym that stands for Encyclopedia of DNA Elements. And that program was mapping functional genomic elements in the genome. So you're probably familiar with genes, but there are other parts of the genome like enhancers, promoters, and other important sequences that sort of direct those genes and tell them what to do and when to do it and where to do it. So those kinds of consortia, GREGoR, the one that I lead right now, is another one like that, have a lot of phone calls because we have a group of researchers at different universities working together to solve a specific problem. So we have calls where we talk about like, how are we going to format our data all the same? What are some of the collaborative projects that we want to do together? Right now, GREGoR started about a year ago, so we're doing a lot of like, what are our policies? Mm-hmm. What are our standard protocols for things? How are we going to do X, Y, and Z? So we do a lot of phone calls like that.
Standardization is probably the most underappreciated thing we do in science.
It is, and it takes a long time. And it takes even longer if you don't do it from the start.
Yeah, exactly.
If you have to go back and fix it later, like that's going to take twice as long.
And two conventions when they like, we have in electrochemistry, we have European versus American.
Right. Oh,
and
that's
like,
got to be challenging.
Do you graph negative on the left and positive on the right, or positive on the left, negative on the right? And it's not standardized in any way.
So we have to do the same thing. Imagine for cataloging a phenotype, information about a phenotype. So even something as simple as like what someone's blood pressure is. There are different ways that a clinician could measure your blood pressure using different equipment. There are different ways maybe that they could report the data. Even something like male, female. I remember in a program I used to lead, we had some people wrote M, some people wrote F. Some people wrote male. Some people wrote 1 or 0. And that makes for a data set that you can't use together and you can't use with any other data sets. And the real value in any large data set or any data is like combining it with other data.
Yeah.
So you want to make it as easy to do that. So that's like another big thing that I do is have these consortium calls.
I mean, presumably that's becoming more important with things like machine learning and AI, which is probably infiltrating your world as with the rest of us.
Yep.
Standardization is everything.
Yeah.
Excel spreadsheets with labels. Who knew how valuable those are?
Right. So we do that. And then the other thing that I do is have a lot of meetings with my colleagues. Meetings where we talk about the applications that we're considering funding. Meetings where we talk about how we're spending our money, meetings where we talk about the meetings that we're going to have later, the meeting to plan the meeting, which is one of my favorite genre of meetings.
Yeah.
So yeah, it's an administrative job. I don't do science anymore. I mean, I used to. I was a graduate student too. I studied exon activation. I was a postdoc as well. I see somebody in the chat saying that they were a postdoc at NICHD, which is great. I was a postdoc at NIEHS, which is the Institute of Environmental Health Sciences. So I did used to work in a lab, but I've been in administration now for almost 15 years, I think. 14 or 15 years. So I don't actually do the experiments anymore.
If it makes you feel better, I don't do them either. I mostly do administration. I write stuff all day. That's my job.
And I honestly don't miss doing experiments. I liked some aspects of it, but you know, some aspects of it are extremely frustrating.
Monotonous and
I don't
terrible.
have to do that.
Yes.
Thank you, Gigi.
Wow. So a fun factoid, I didn't know this, but Twitch just flagged NHGRI. And it's presumably because it's some iteration of the N-word.
Oh, well.
So I have to approve this message before it'll post.
Oh.
Hey, wow.
Okay.
Heads up.
That's
you
awkward.
might want to notify the NIH about that. Twitch is flagging their messages. Wow. Good to know. All right. CJ said, I was a postdoc and later a research fellow at the NICHD, which was acceptable.
Yeah.
And I think it's so cool that you are here. I can't imagine my old PI doing something with Twitch. Oh, you'd be surprised. We've had a lot of people on, a lot of diversity. I mean, kind of got to be fearless to be willing to play video games and talk science at the same time.
That's true.
Like you're on public record just embarrassing yourself in games sometimes. And
Yep.
All right. Calm Down Bronco wants to know, what drove you to want to work for the NIH?
Okay, that's a great question and one I love talking about. And I really hope that this is going to get me... Wait, why can't I get through that?
A rocket.
Isn't
Right?
this a rocket?
Is that-
Oh, okay. Okay, what's this?
Is that the bomb? It is, isn't it?
So,
Reese's
hope
Pieces,
so.
where are you? We need your help.
oh, because I feel like I would press B to... What did I just get? I don't know. Okay. Long beam. Oh, that's good. That's good. Yes. Okay, great. Perfect. Okay, so...
So I don't know if you've seen this anytime recently. There's the map.
Oh. Wait, pause.
Let me see if you guys can see it too. Yep. So there's the map for Metroid.
Surprisingly...
Yeah, it's got some depth to it.
Okay. All right. Okay.
That's intense.
Okay. All right. So how did I end up at the NIH? So I was a postdoc, like I said. I planned to go into academia. That was always my goal. I love genetics. I worked as a technician in a genetics lab identifying rare disease genes, actually, before I went to graduate school. And I just really liked everything about it. It seemed like a great job for me. But when I got to my postdoc, I ended up going to a lab which, like I said, I was a postdoc at one of the NIH institutes. My PI was great. He was a really great mentor to me, but like I just was not that excited about the research I was doing. And when you're gonna go apply for faculty positions, you have to be at that career stage really excited about what you're doing and doing your best work, right? And I also had some personal things happen around that same time that kind of threw me off of what was happening. And I just sort of lost the drive during about the second year of my postdoc to go into the job market. I also am married to another scientist. He's further ahead of me in his career, so he already had a research track faculty position. And I just couldn't see how both of us were going to do this at the same time. Like, competing for funding is very stressful. Running a lab is very stressful.
Yep.
And
the divorce rates those first 5 years are astronomical.
I'm sure.
They've done research. I don't know if it's NIH funded, but they've done research on it.
No, I can believe that.
Yeah.
So it just didn't seem, for so many reasons, like the right thing to do. So then I started to think, okay, well, now what do I do? I graduated my PhD in 2005, I think. You have to check my biography. I can't remember.
I will look up your file right now.
Pretty sure it was 2005. And when I was in grad school, they didn't really teach you about...
2005, Case Western University.
Yes. Now, what am I just don't think I can do anything there. Yeah, so they didn't really talk to you about any other sort of career paths. And there was, I think, a lot of, I don't want to do that.
First.
Okay, fine.
Or clip through the floor. It's up to you. Just a minor comment on that. The reason we don't talk about any other career is because most of us don't know any other career than academia.
That's true.
It's one of those things that it's perceived as like academia is the pinnacle of scientific achievement, and it's not true. It's a complete fabricated lie propagated by people that work in that area.
It is
self-grandizing.
That's yes. It's great in many ways, but it's not for everyone. And I would say that when I was a PhD student, and I'm sure now, but I think that PIs are a lot better about being encouraging of people doing what is right for them and not what they want.
You can no longer just bully students, literally beat them up.
They used to do that.
Yeah, no, exactly.
So anyway, I didn't know anything about any other career paths, and I sat there and I was like, okay, well, what are the things that I like doing? What am I good at? So at the time, I was doing a postdoc that required me to do a lot of microscopy. Pretty sure I was in that room already. So.
Do you want the map?
Well,
I don't think it'll help.
No, it's not.
No,
I've heard this is going to take me to that elevator. No. I had been sitting in a room by myself on a microscope for hours. And that is interesting. And I created a lot of really pretty pictures of chromosomes.
Yeah.
But I like talking, and there's not a lot of people to talk to in a dark microscope room.
Yeah.
So I thought I wanted to do something that was a little bit more customer service centered. So I applied for things like running sequencing core facilities, or working as a consultant, helping people design genetic analysis studies. And I thought about going back to school and getting a degree in genetic counseling, because that's an area where you sort of, now this is where I first came from.
So I think I found you on the map.
That's like, this is way at the beginning.
So I think you're here.
Yeah.
I don't know where that's the beginning.
Yeah. Do I have to go down?
No.
Oh no, that's where that's like.
Isn't that the end?
That seems endy.
Yep. It's not there. Right there, start, you're here.
The problem with this game is that it all looks the same.
Yep. So we can go up and left.
I haven't been in there, but those are Metroids.
No, you got the long beam.
Oh yeah.
So you got that one. Further up.
Okay, while you're finding the bombs, I'll keep talking about this. So I was going to. I thought about going back to.
Reese's Pieces. Go orange column, top right.
Okay, I was in. Okay.
What is that? That's a rocket. Orange column, top right.
Varia is what I just got, isn't it? Is that the?
So that's a boss.
Oh no, I don't have that yet. That's the plasma thing that goes.
Yep. Here, I can show you guys what we're looking at. Sorry, we got important stuff to do here, ladies and gentlemen. What is that? Is that something?
That's an energy tank.
Okay, doesn't help.
I think we're just gonna keep
grinding.
Yeah. Okay.
Maybe try the column top.
I'll try.
So if you go up this and go to the top right door, that takes you towards orange.
All right.
We'll figure it out from there.
Okay. So genetic counseling is a very interesting career where you people get genetic testing all the time from their doctors, and the doctors are not geneticists, and they don't necessarily know how to interpret the results or how to tell you what that means. And a lot of genetics isn't super simple. It's not like you have a mutation that means you 100% have you're 100% going to get a disease. But you still need to make some informed decisions about what you're going to do. And so genetic counselors help that. So that seemed interesting to me, sort of bringing genetics to a more lay audience. But like I couldn't imagine going back to school after I had already finished a PhD and done a postdoc. So working as a postdoc at the NIH, you would get job postings emailed to like our all hands listserv, and a program officer position with specific expertise in epigenomics, which is what we call the sort of layer of regulation on top of DNA that tells genes to turn on or turn off. Things like DNA methylation, histones, histone modifications. Anyway, I had that expertise as well, and my PI, who knew what a program officer was, said, "Oh, you'd be really good at this, you should apply to it." And I was like, sure. I really had no idea what a program officer did, but I applied, interviewed, and the rest is history. And then I was a program officer at NIEHS, the Environmental Health Institute, for almost 10 years before I moved to NHGRI about 4 years ago.
Reese's Pieces. How many scientists does it take to beat a Nintendo game?
Like, clearly more than 2.
No, see, what we need is the ASCII art and 30 pages of text.
Yeah.
You know, old
Exactly.
school. You want to know what the speedrun record in this game is?
It's just gonna make me sad.
What's your guess?
It's probably like 23 minutes.
Metroid any percent is 9 minutes and 12 seconds.
I've barely even gotten up and down this column. Oh, no, this is at the start. Okay, so where's the orange? I gotta go over to the right somewhere.
We got this. Sorry, I'm distracted.
No, it's fine.
Yep.
Everyone's enjoying watching me.
If you go to the right, go up.
Oh, I saw some orange on the side there. Oh.
So if you go up,
Jeez.
you're down here on this column. If you go up all the way to the right.
Not here.
Are you're down here probably?
No.
You're not in that column anymore.
For no help.
I'm not. Yeah, there we go. Okay, I know where you are. You're right here.
Okay.
So if you go down
Another
to
level.
the right,
Okay.
yep.
All right.
That gets you to the orange column.
Okay.
And we're gonna go up and right because Reese's Pieces told us to.
Okay, great. Thank you, Reese's Pieces. Oh yes, imprinting. Yes, that's an epigenetic phenomenon.
Drosso killer. Drosso, is that a shorthand for Drosophila? Is that slang?
I hope it is. I'm assuming it is.
Yeah. I mean, they followed when they learned you were the guest, so
Yeah.
presumably they know. Drosso says, "You guys should try Fall Guys if you haven't already. I've been addicted to it since they made it free recently."
I've
Have you
never
seen
even.
Fall Guys?
No.
It's a very cute, like, like combat survival game. I don't know how to describe it, but Drosso, I've seen a lot of it. I haven't played it much, but it does seem like a really fun game. The dirty secret about the Ask a Scientist gaming is most of our guests haven't played any of these games in like 30 years. And asking them to play a new game is a non-trivial ask.
Now, is this it? Oh, no, wait. Oh, gosh. Oh, this actually, I feel good about this.
So this is going green.
Yeah. These flying paramecium things are something that I have the capability to beat, so I don't know why I keep getting the energies. Oh, okay.
All right, so time flies. We're 40 minutes in. We should probably do one of your predictions. Which one do you want to do?
Oh, well, let's talk about, well, this is a good starter one.
All right, we're leading into genetics gently.
Yeah.
All right, ladies and gentlemen, it is prediction time. So If you're not following us, click the follow button. It's going to be a chance for you to spend the internet points. We call them standard internet units on this channel. They're useless for anything outside of this channel, except you can make us take a drink, or you can request a factoid, you can unlock an emote, but you can also gamble on science questions. So usually predictions are used for gaming. On this channel, we use them for science. So if you're not clicking follow, click the follow button right now. You'll get 300 standard internet units, and you can gamble them on questions. And the first question of the night is, almost all calico cats are male or female? So we're always surprised by the outcomes on it. So I'm looking forward to seeing how this one unfolds.
I'm going to predict that this is a softball.
So for those of you that haven't done this prediction before, click the predict button on the top of the chat there, and you can gamble as many points as you want, depending on how confident you are in the answer. Note that you are on Ask a Scientist Gaming Honor Code. You cannot cheat. You have to use it with your own knowledge. This is probably one Google search away, but we're asking you to be honest in your answering of the question. So we have about 1 minute and 15 seconds. Almost all calico cats are male or female.
So, okay, I have to pause this for a second so that I can see what is going to happen here.
What, in terms of what?
Like, how are we going to see what people picked?
I'll show you right after. Yep.
Great.
Yeah. No, it's fun to watch.
Okay.
So they have about 1 minute left to answer this question.
All right.
But in the meantime, almost PhD done to ask, did most of your colleagues complete a postdoc before working at the NIH?
Yes, almost. Well, people in the same kind of job position that I am, program officers, not only did many of them complete a postdoc, but they also were faculty. So in general, you have to have a postdoc. Now, one of my colleagues that I work with the most actually doesn't have even a PhD. But in general, you do want to have a PhD and a postdoc.
All right, 30 seconds left. Almost all calico cats are female, male. It is 100% female right now. Somebody dark horse bet. Somebody put male in there. Bet 10 points. It's going to be like a 30-to-1 payout or something ridiculous. Someone commit. Or they just all know the answer in that answer.
I know, Oh, this is energy, which I don't need. I've got so much energy.
Infinite energy, actually. All right, Lisa, what is the answer? I think we already know.
Well, how many percentage of people are correct? Yes, they're pretty much all female. And that is because the orange and black coat color genes are on the X chromosome. And in female cats, or in female humans, pretty much all female mammals, one of the X chromosomes gets turned off in every one of your cells. And so in some cells, the chromosome that has the orange color allele is active. And so those ones have orange fur, or those cells have orange fur. And in the other cells, the black is active.
I found it. Keep going right.
That is actually what I did my PhD on. Not cats, but exon activation. Okay, keep going right. Yeah, that's what I thought. Once I saw these guys, I was like...
So you went through the green. Where did you end up?
Yeah, that looks like where I am.
Right, so...
I'm in another orange. It's okay, I haven't been here yet, so I've got...
Congratulations to anyone that said female. You have pride, but no additional points because nobody bet against you, so... that was a unidirectional.
I'm really glad to know you were all paying attention in genetics class. Kudos to you.
Exactly. We'll take that. So where are you? You're inside the...
Yeah, but I just got here, so this feels promising.
Yes.
Good.
So if you go left...
The thing about talking about science while playing this is that I can't keep that mental track of like, okay, I went in this door and I did this, and then I went in this door and I did this.
Yeah. No worries. It's just for fun.
Yeah. It's
but
never
anyone,
just for fun.
Yeah, exactly. And pride. All right. Green Jenny, thank you for the follow. Sorry I didn't acknowledge that earlier. We were tied up in something, trying to find a bomb, presumably.
Yeah.
But yeah, thank you for following us. Enjoy your standard internet units. Make us drink alcohol. Request factoids. Wow. So Lisa's about to get a bomb. This is exciting.
Oh, God.
There it is.
Thank you.
There it is.
The world has opened up to me.
Ken is a good navigator. Oh no, this was a disaster. All right, so the question is, you got bombs. Do you want to keep going or do you want to play something else?
So, is this going to just sit here in the background?
Theoretically, we could.
Okay, let's do that.
What do you want to do?
Well, let's play Dr. Mario.
Dr. Mario? All right, so press pause on that guy. Actually, I don't know if we can run two Nintendo.
Oh, well then should we play Wii?
Yeah, let's do that.
Switch?
Yeah,
Right.
let's do that.
All right.
So let's pause this guy. I'll move your chat so you can see it.
All right.
All right, we're going to switch over to the Nintendo Switch. This should be a fun game. This one, you can do anything while you're playing it. Wait for that to load. Maybe it powered down.
Oh. Yeah.
This one needs significantly less maps than Metroid did.
0 maps. All right.
All right. So this game, you might have to introduce this one.
Okay.
This is a weird one.
So it is a weird one. That's why I picked it. This is Katamari Damacy. And you are this little guy. I can't remember what your name is. And you have this big sticky ball that you basically roll things up into. And this guy who is like the emperor of all galaxies, I can't remember what his name is.
Something like that.
He tells you what size he wants the ball to be. So I need to roll the ball to at least 10 centimeters here.
All right. So what's your guess on the speedrun record on this?
Well, I don't even know how you... I don't know. That has to be like on the order of hours.
27 minutes.
What?
It's always less time than you think. All right, let me show you the chat.
All right. Oh, the king of all cosmos. I was close.
Oh, we're learning the lore right now.
Wait, why am I... So I used to play this a lot, but on a different game system, and I was a lot better at the controls then, and now I'm a little not great.
So this is literally just snowball garbage until it's bigger.
Yeah.
And it scales beautifully as you go. So right now you're picking up...
Oh no, 50 centimeters is where I need to get to. I'm sorry, I started at 10 centimeters. My Katamari was 10 centimeters. And now I've got to get to 50. This is probably going to make everyone nauseous watching this game. Sorry,
Anyone
everyone.
getting motion sickness? It's a hypnotic game. I loved watching a friend play this. Like, he was really good at it. 'Cause you start rolling up planets. And,
Oh yeah, you get
huge.
Yeah. Eventually I'll be able to roll up that squirrel.
Yep, and
Not
cows.
right now.
Katamari. All right. Here's the title of the screen.
I always like to see, now that we're all on Zoom all the time, like you can see people's looking at files on their computer face or reading face.
Yeah.
And now everyone can see my intense game face, or when I was playing Metroid, my trying to jump out of the way of things face.
And the occasional yelps. And this game aged well. This came out in, let's see, I looked up all the dates on these. So this was 2004, so almost 20 years later. Yeah. But this one's easier to play while talking to things.
It is.
Or presumably it is. All right, Calm Down Bronco wants to know, what are some of the rare disease genes you are researching? What makes them rare?
So we actually in GREGoR are working on so many different diseases at once I couldn't even tell you, right? What we do is we work with clinicians and collaborators all over the world who have gotten together samples from people, DNA samples, from people with rare genetic diseases that they're pretty sure they have some evidence are heritable and probably caused by one gene or a very limited number of genes. And then they sequence them to try to find the parts of the DNA that are different, that are likely to be the causal mutations. And then they do all kinds of other sorts of studies to convince them that they're right. Like, so they want to make sure that if you put that, see, now I can roll up birds.
I'll be back for you, dog.
Yep. Yeah. I can definitely get that animal, that mouse. So GREGoR is, I would say, I just can't even remember. We had a program before this called the Centers for Mendelian Genomics, and I want to say that they identified the causal gene for like 300 different genes or different phenotypes every year, right? So it's not just one. I personally, when I was a technician, worked on mapping a rare genetic disorder called mucolipidosis type 4. And back in those days, before the Human Genome Project was finished, and before we had the human DNA sequence at our disposal, it took a long time to do that. So it took several years to even find out what chromosome the gene was probably on. And then you had to find out what sort of part of the chromosome was it on. And then you had to find all of the genes in that area because you couldn't look it up. We didn't know what the genes were, so you had to figure that out. And then you had to sequence them all. It took forever. I think the first human disease gene that was mapped was Huntington's disease. And it took them 10 years after they found out what chromosome it was on to find the actual gene, and now we do it really quickly.
So how do you differentiate a single point mutation from like an epigenetic or a?
So actually that's an interesting question. So let's imagine it is just a single point mutation, which a lot of them are. What you do is you compare people who have the disease and people who don't have the disease.
Okay.
And you look for variants that are different in those two populations. So often in these kinds of studies, we're looking at the parents and the child. Parents don't have it, but they're probably carriers.
I see. So most of their DNA is similar except for...
Yep.
Yeah.
And then you look at the child and you're looking where the child has like 2 mutations, but the parents only have 1, if it's a recessive phenotype. So, and then you have to say, okay, now is this variant, what kind of thing does this variant likely do? So you look at where it is in the gene. Is it in a part of the gene that looks like it could be functionally important? We have all kinds of like software to predict what the structure of the protein is going to be and what different parts of it are going to code for like a DNA binding domain or a part of the protein that's going to interact with another protein that's going to be important. So you can tell this is probably an important part of this protein or not. And then you also then compare it to other people in the population who don't have the disease. And you say, is this a common variant that a lot of people have, but they don't have the disease? Oh, good, I finished. Or is this only specifically found in people who have the disease? So that's sort of largely how you do it. But we got fairly good at doing that pretty quickly. And so if you have a genetic disease and we think it's a Mendelian sort of simple inherited disease, you can go and have genetic testing. They'll do something called exome sequencing, which is basically they select only the gene parts of your DNA, the coding regions of the genes. And they sequence only that, and they say, okay, well, here are all the important parts, and here are the mutations that we found here. And so this is likely to be a causal mutation. But over 50% of the time, we can't find anything in those seemingly important regions. So that's what GREGoR is doing. We're trying to figure out how do we sort of operationalize identifying the causal mutation when it's not that simple. So sometimes that could be an epigenetic kind of change, like a methylation mark that turns a gene off when it's supposed to be on. And we have to use different kinds of assays to see that. or sometimes it could be a mutation in a part. Oh, that dog got you.
Take that.
Now I'm gonna go get these ladies. So it might be in another important functional part of the genome, like an enhancer or a promoter. So those are the parts of the genome that tell genes when to be expressed, how to be expressed, where to be expressed. When you're looking at stuff like that, it's like a lot more DNA and a lot more potential variants that you have to sort through. So it's just harder to do than it is when you're just looking at exomes, which is like, I should know this, what proportion of the genome is coding regions of genes, exons, but it's really low, right? Most of the genome is not that.
If anyone knows, throw it in chat.
It's like, I want to say it's like single digit percentages. So it's challenging to do that, and so that's what we're trying to do in GREGoR, is develop better ways, new ways to think about approaching those cases that we can't solve just by looking at in the most obvious places, which is the exons.
All right, CJ has a specific one. Any idea if the T2T genome build will be incorporated into NCBI?
Wow. Thank you for asking me about that.
So explain what those words are and
right.
abbreviations. Very specific.
That's a great question. I was hoping someone would ask me about that.
Is that?
Okay, so here's some background for people to know what T2T is.
Mm-hmm.
So you might remember back in 2001, we said, hey, we've sequenced the human genome for the first time, and here's the draft sequence of the human genome. And that was like a really big deal. When you were working in an area where you needed the genome, like it changed everything about how we approach things. It made things so much simpler.
Should we throw this prediction up there now?
Oh. Not yet.
Okay.
But almost, yes.
All right.
Okay. So, but this is one of my factoids.
All right.
So, that was a really huge achievement. And then it took them like another 2 years and they kind of cleaned it up and made it better. And that became like the first like real reference genome that we used. But that actually didn't sequence the whole genome. It sequenced, I think, like 92% of the genome. And the rest of the genome just got skipped. And so if you went to like the NCBI genome browser, or the UCSC genome browser at the time, which I used a lot. Now, how do I get to the world again? This is not where I want to be, but here are all the other guys that I have gotten.
You know, the space mushroom.
Exactly. Those parts of the genome that they skipped were just hard to do, right? They were in repetitive regions of the genome. And the way that we sequenced the genome the first time is you basically broke it up into a whole bunch of pieces. You cloned it into different sorts of vectors so that you could replicate that region to sequence. And you would sequence very short pieces of DNA, and then you'd try to assemble it like a puzzle. But the pieces of DNA were really too short to assemble the puzzle if they were too similar to each other, right? And you didn't have enough polymorphisms or those like DNA changes that would help you understand, well, this one came from here. This one, even though it's very similar, came from somewhere else. So for a long time, we just like lived with that. And those parts of the genome that were not sequenced, some of them were really important parts of the genome, like centromeres, which are made up of a very specific kind of DNA repeat. And other parts that had things that we knew were probably important, but we just couldn't do. And so the T to T is, that stands for telomere to telomere. And that is basically the first gapless genome reference. That sequenced the whole thing from telomere, which is one end of the chromosome, through the centromere all the way to the other telomere.
That's awesome. So that's nerd speak for end to end.
Yeah.
That's awesome.
Right.
Today I learned what T2T is.
Yes. And so, yes, that was actually done, that was funded by NHGRI. One of the lead investigators of that program, Adam Philippi, is an intramural investigator at NHGRI. And one of the other ones, Karen Miga, who's at UCSC, actually, Karen and I went to graduate school together. So we are trying to get that incorporated as a standard reference genome. And I'm sure that you will be seeing that in places, more standard places. But what kind of has to happen now is that we need to make the tools, we need to put all of the information that we already have on the existing genome builds that we use onto that one. Because a genome reference is really only as helpful as the annotations that are on it and the things that you can do with it. So that's something that we're currently working on doing. And then you have to sort of slowly get scientists to change what they were doing and move to a new reference. So I don't remember this one. But now I'm getting sushi. So yeah, that's a great one. That's going to, I think, be really helpful for a lot of things, including rare disease gene diagnosis. You can ask me about the pan genome next if you want. I can ask you. Whoever asked that question.
That's awesome. Yeah, thank you, CJ, for that. So we're an hour in. Anyone just joining us? Guest today is Dr. Lisa Chadwick. She is a NIH program director. She knows a lot about genetics, genomics, GREGoR Program. I can't remember the other one.
NHGRI. That's the name of the institute I work in.
So if you guys have any questions, throw them in chat. All right. CJ has a related follow-up. I'm going to let you read this one because there's acronym soup in it.
That's basically
Edger versus
okay, I don't know
Desquat. Desquat. I'm going to say it's Desquat.
I don't know what those are. I'm sorry, PJ. I don't do the experiments anymore.
Look it up.
Those are definitely analysis programs, I'm sure.
I just searched for Edger and I got best lawn edgers of 2022. So
right.
I've given up on that search.
I'm sorry, I can't answer that question.
They are RNA sequence analysis programs.
Oh, okay. So I'm not a computational person. I do transcriptomics is part of my portfolio, but my part of it is more the experimental side, technology development in transcriptomics, and different assays to learn different things about RNA. I have a colleague who is a program director who does more of the computational side of that. So I could phone a friend, Dan Gilchrist, but unfortunately...
Does he play video games?
He does. Well, he's got children about the same age as mine, and I know they play video games because I've talked to them extensively about Breath of the Wild.
It's a classic. Yeah, David Collins played Breath of the Wild on screen. All right, Compound Bronco wants to know, do you have to sign an NDA-style contract when dealing with NIH grants?
So, yes. Well, not exactly, but sort of. So it is confidential. Everything that I see in grant applications is confidential. I have to log into our internal systems to see that. I have to be granted that level of access. I also had to have a background check to have my job, like an extensive background check. So we can all talk about those things within the NIH, but I can't talk to other people outside. Not within the entire NIH, but I mean people who need to know. Mm-hmm. But no, it is confidential. And in fact, your grants, when they get reviewed, they're confidential as well. The people who are reviewers have to sign different things that say that they're going to protect the confidentiality of the applications that they read. But yeah, so I don't remember signing anything in particular, but
yeah.
I probably did.
But you get the note every single time by
you do have to agree to exactly.
Yeah.
Yeah.
And it's the same with reviewing papers. Like you'll exclusively look at this, not share it with anyone.
Right.
Yeah.
And it is kind of a fun part of the job because I do get to learn about things before they even come out to anybody else.
Yeah.
We do a lot of technology development and sequencing at my institute. We fund small business grants. Well, all of the NIH does. And so there was recently like a big sort of revolutionary new sequencing platform that was announced from a company called Ultima Genomics. I think they released that at AGBT this year. Anyway, I've known about that for a couple years. And so it is fun to see stuff before it becomes public.
That's an awesome part of the job. You also deal with the rejections and the crappy proposals too.
Yeah, I do.
I gotta imagine one of the hardest things. So NIH, NSF, you're much more restricted in what you can fund than, say, the Department of Defense, where program officers essentially have full control over what they want to fund.
Yeah.
I mean, you've seen proposals that you want funded that did not get the rankings.
Yes, I definitely have.
Like that.
It does.
I mean, you obviously have to deal with it, but like, how often do you see that happen? How often are the wrong people getting funded in your eyes?
Well, so I trust the reviewers and the advice that they give us in rankings. So I know that they're doing their best job, and they're trying to help me find the most exciting, most innovative, most promising science.
Fair.
But yes, there are things that come through that I would love to have seen done better, or and to have gotten fundable scores that I just can't find.
Yeah.
So what I do is, hopefully those people, I need to find a new place to go. Where can I go? Hopefully those people call me when they get their score, and I can talk to them about, I'm going to fail this level. I can talk to them about how to make their application better and how to be more successful the next time. That's like one of my favorite things in my job, is to see someone go from having a grant application not discussed to getting funded. So that's basically what I would do. But yeah, I do have to deliver a lot of bad news.
Yep.
I talk to a lot of people who are disappointed and frustrated. I call our job sometimes therapist to frustrated researchers, because sometimes you get the summary statement back and you're just mad, and you're allowed to be mad about it. And I'll just listen to you and then try to help you think of constructive ways to move forward from that.
I mean, but the general advice I've heard is, when talking to program officers, program directors, spend more time listening than saying what you want to say, because their advice is more valuable than your opinion, because your opinion obviously didn't get it funded. So listen to the feedback.
Right. I always say, like, I didn't spend months and months of my life writing that grant. I'm not personally invested in it, and so I can read the reviews and take that feedback much differently from the way that you can. And sometimes people just read something in there that makes them personally upset, right?
Yeah.
And that's not always the most important thing, and so I can help sort of sort through what's important for you to think about and what's not.
And sometimes the reviewers just flat out get it wrong. Either they have a misunderstanding, or
they do sometimes. But what I tell people is, that means you didn't explain it well enough.
Yep. And that's the usual way I deal with rejection, is try to be introspective as possible. How could I address that? But there are some that are just terrible. Yep. And there's nothing you can do about those. But program officers tend to know which ones those are, and which are nonsensical and off the wall. But all right, we should do a prediction.
Okay.
Which one do you want to do?
All right. That one.
This one.
Yeah.
All right. So it is time for another prediction, time to spend some standard internet units. If you're not following us, click the follow button, get your points that you can bet on questions, make us drink alcohol, and or request a factoid. So I modified this one slightly, but I think it's the same question. I did an over-under instead of two different values.
Okay.
All right. That is an angry child in the background.
Yep.
All right, we're going to do a prediction. Click the follow button, and now click the predict button. Put your money where your opinion is. Ask a Scientist Gaming honor code. What is the cost to sequence a human genome? That is the current cost to sequence a human genome. Is it greater than 10,000 or less than 10,000? Put your predictions in there now. Bet as much as you want based on your confidence, or just bet for fun. It's totally up to you. These standard internet units are useless outside of the stream, so spend them as you as you feel fit. At what depth?
Oh, okay.
CJ, dropping bombs.
I don't know.
That's a good depth. Presumably means
but
precision.
CJ, you better get it right.
Yeah, that's all that we have to say. Reese's Pieces redeemed. Take a drink. Lisa, cheers. Thank you for joining us, everyone. Hopefully, you have a drink in your hand on a Wednesday night, hanging out with us playing Katamari Damacy while talking genetics and genomes and sequencing and the NIH and grants and funding. Cheers, everyone. Thank you for joining us. Cost to sequence a human genome. Yeah, without revealing things, we'll talk about it after this one's done.
Okay.
At what depths? CJ, you know too much.
Yeah,
It's general audience questions. We should do one really technical question every stream that no one will know anything about.
Well, I'm an administrator now, so that will include me. I won't know the really technical.
Oh no, you do something specific. What's the success rate of funding in your program?
I can tell. Yeah, I actually.
I mean, that'd be a fun question, actually. Someone request a factoid. With the money you win from this question, what's the cost to sequence a human genome greater than 10,000, less than 10,000? You have about 10 seconds. Throw your answer in there. Throw your prediction. All right, and it's closed. So Lisa, if you want to see the...
How do we...
So the betting was heavy towards greater than 10,000, but the number of people did less than 10,000. So what is the answer?
It is less than 10,000. Actually, it's significantly less than 10,000. It's under 1,000 at the moment. You can go to the NHGRI website where one of my colleagues has been diligently tracking the sequencing costs from the various genome sequencing programs that we have at the NIH. And it's actually right now, it's like around $750 a genome.
Jeez. So your little note on this, $300 million to sequence the first human genome in 2001. Today it's under $100. So is that
under 1,000?
Or under 1,000, sorry.
Yeah.
And that's following like Moore's law, right?
That is following Moore's law. Exactly. Well, actually, so if you go to our website and you look at it, there is a graph that you see a lot when you see people talk about the cost of sequencing a human genome, which really probably only CJ has looked at. But it shows that where it followed Moore's law, and then when there was a major shift in the technology that we use to sequence DNA, there's a big change from the standard method, which was called Sanger sequencing, to more automated methods, or not automated, but totally different kind of way of doing it. That changed the price significantly. So you see it takes a big dip there.
So GreenWort, a spreadsheet I helped on many times.
Yes.
So someone has seen it.
Yep.
I mean, so the question from CJ about at what level? So is this every single base pair sequencing, or is this?
No, it's basically how many times. So you say things like 10x coverage. That means every position in the genome is represented roughly 10 times. That just is the denser, the more coverage you have, the more sure you can be of the results that you get. Especially finding those rarer variants. Sometimes sequencing has errors. Mm-hmm. And so if you don't sequence at a high enough depth, it's hard to know whether the variant is real or whether it's just an artifact of sequencing. But if you've seen it in 28 out of 30 copies of that sequence, you're pretty sure it's right.
And so what's your general opinion of the public, like 23andMe and the family history? And
I think it's really cool. What I like about it is it's gotten many more people interested in genetics.
Yeah.
And, you can learn like cool things like, I have a 23andMe account and I love reading the little histories or the trait reports that it gives me. Like, are you likely to have curly hair or straight hair?
Yep.
Are you likely to have a widow's peak? Are you likely to be a super taster? That's just like a few. There's all kinds of trait reports that you can get. And I like that it has sort of democratized genetics and opened it up to a lot more people.
So how paranoid should I be about insurance companies having this information?
So we do have laws about that. The Genetic Non-Discrimination Act. Now, do the laws probably need to change with time and the amount of information that we're able to get about people? Probably. But I'm not super worried about it myself. Maybe I should be more worried about it.
Yeah, companies follow the laws, right? It's fine.
Yeah, it is really tightly regulated. Yeah. Any sort of human subject kind of research is very tightly regulated. So I think it's really important to read the consent forms when you sign up for anything like this so that you know exactly what you're opting into. For example, I know with 23andMe, you can opt into all kinds of stuff, sharing your data in all kinds of ways. And I did that because I know it's important for researchers to have access to genetic data to test hypotheses and generate new hypotheses. But you don't have to do that. So you kind of have a lot of control over... Okay.
Oh, nice.
You have a lot of control over your data.
A lot of this game is getting big enough to find a new area, right? And knowing the map.
Yeah, but I've gone back to being smaller now. I've already rolled up these birds before.
Yeah.
And now I'm much smaller.
Yeah. Oh, calm down, Bronco. I didn't even see. His question is, how do services like 23andMe work? Are they looking at SNP specifically or more than that?
So I think that they... Okay, let me just say I actually don't know exactly.
Is that proprietary or it's just...
No, they probably say, but they probably have specific panels that they run, so they're looking at, probably not the whole genome, but specific regions. So kind of like I talked about exome sequencing before, which is only looking at the coding regions. They probably are running a panel that's really looking at the parts of the genome, mostly that they know that they can tell you something about. But they're also getting a lot of other sequence data alongside of that they can use for their own research studies that you can opt into. SNPs is a single nucleotide polymorphism, so that's just one letter of your DNA that's different. And that's sort of the basis of a lot of the sort of genetic studies that we do. And a lot of those things are a little bit more complicated, so that's what makes genetics challenging to interpret. It's not as simple usually as one gene causes one phenotype. It's one allele of this gene and a different allele of a protein in another gene that's a protein that interacts with that protein. So those kinds of sort of more complex calculations might be proprietary, how they do it. Some of that is more challenging.
Yeah, Sarah and I spend a bunch of time talking about that, a lot of it's probabilistic, right?
Yes.
If you have a marker, but it's going to become increasingly important with in vitro fertilization and screening of embryos, and now with the Roe v. Wade overturn, that becomes really muddy. laws need to be updated. There's
Yeah.
no question about that. But if you can have a slightly taller kid, one with a slightly lower probability risk of cancer, like, do you do it?
This is not my best Katamari game here. I'm really not doing
No
well.
worries. CJ has a follow-up. Are those genetic testing companies subject to IRBs, or because it's commercial product, now are they exempt?
No, I think they are.
They have to do IRBs.
Yeah.
That's interesting. So they have
I'm pretty sure they are.
Huh.
I just...
No.
I'm supposed to get to 1 meter and I'm at 19.
Yeah, don't worry, we have a rage emote. That's from Aaron Vanucci when he quit Madden in 2007 at halftime when he got an interception. No worries. All right, Calm Down Bronco wants to know, if I apply for a graduate grant or fellowship during my PhD, is the pre-submission consultation available to me?
Absolutely. So every NIH institute has generally 1 or 2 people who are like the central program contact for fellowships. And so if you look in the F32, that's one of the fellowship mechanisms funding announcement, there's always a section in the funding announcement that's to scientific contacts. And that will bring you probably to a list that will tell you who the right person. Yeah, I know. I'm sorry. I'm sorry.
So
really
aggressive. I'm not mad. I'm just disappointed.
Yeah. Yeah. Okay, so you contact that person, and then a lot of times they can put you in touch with another person, another program officer who is like in charge of that sort of scientific area, because we kind of split grant applications based on different scientific areas. So you absolutely should talk to somebody before you submit your application, even for fellowship.
So we're a little past, but Calm Down Bronco requested a factoid.
Okay, great. I already used some of these. Okay, so we've talked about the different kinds of grants that you can get, and one of the big ones is the R01, which is like sort of the bread and butter grant of researchers who get funded by the NIH. So what is the average age of a PI or a researcher when they get their first R01? It is... Oh, I tell them now, right? Or do you want to guess?
Kind of want to guess.
Okay, go ahead.
Say average age. Average.
Got my post.
We'll say 38.
It's in 2021, it was 44.
All right. I mean, some people don't even apply till they're 60 for the first time, right? So it's going to perturb the average, but that's interesting.
How are they funding their research until the age of 60?
NSF?
Oh, that's true.
Yeah,
So
you'll get some outliers there.
it's actually a big problem. And so the NIH has been doing a lot of different things over the last number of years to try to make it easier for people to get their first grant at a younger age. Okay, are we going to keep playing this or?
It's up to you. What do you want to
No.
do? No, you're done?
Yeah.
You want to play a goose game or do you want to switch?
No, let's play Dr. Mario.
All right, we'll go down to Dr. Mario.
Okay. Just disappointed the king of all cosmos too many times.
You can't deal with the rejection.
I can't.
All right.
Oh, that means we have to get the bombs again in Metroid, but...
I mean, we can try. I don't know what it's going to do when we have 2 different emulators open. If the controller is going to try to control both, so when you press start on Mario, it's going to do something weird.
I don't know, we'll find out.
Actually, no, we can't because the screen doesn't recognize it. All right, we're getting Mario up. There we go.
Thank you.
All right, Dr. Mario, ladies and gentlemen, which is appropriate for Ask a Scientist gaming stream.
Yep. Okay, I'm going to start with a couple of viruses. All right.
All right, so we got our factoid down. Greg wants to know, this is going back a bit, but I'm still not sure how reviewers get picked for study sections. Could you talk more about how people are nominated and or volunteer? I don't know how you guys do it.
Yeah, yes, and I will say that that's not what my job is. So I can tell you sort of my understanding of it, but there are a different group of people at the NIH who are in charge of putting together review panels. They're called scientific review officers. So basically what they do is they search, like we have all kinds of databases of people who have applied to the NIH and what their expertise is. They get recommendations from program officers or from other reviewers on the panel. But basically they know what kind of expertise they need for their panels based on what kinds of applications are coming in. What am I doing?
You're making it very orderly.
I am. Well, I like to have a nice place to put them.
Yeah.
But like...
No, it costs time, right?
Yeah. Okay, now I can do this. I need to like just
What am
know
I doing?
exactly where I'm putting things.
Well, you're playing video games while talking science in front of a live audience.
Very methodical about it. Yeah.
I'm sure your gameplay style says something about you. I don't know what that is. Get Sarah to do a study.
Yeah.
Psychology related.
Yeah. So, but they're academics. They generally want you to have an R01 already, so that they know that you understand how to write a good grant, and you have familiarity with the process. But we do have a program called the Early Career Reviewer Program, where you can sign up to get put on a panel even before you've had... what should I do with that? Okay. Even before you've gotten your first grant. And it can be really helpful because there's no easier way to find out how to write a good grant or a bad grant than by reviewing a lot of grants. Because you see sort of firsthand what it's like for a reviewer to read grants, and you know what things made it easier for you. So I think it's a really good experience. If you are interested in applying for that, it's called the Early Career Reviewer Program. You can just Google that, NIH Early Career Reviewer, and you can register for that. My understanding is it does take a while to get through that, and that's mostly because they can only have a certain number of early career reviewers on any panel at any time. I want to say like they really can't have more than one on a panel at a time. And the panels only meet 3 times a year, so it can take a while. But that's basically how they do that, I think.
Speaking of careers, have you seen this, the NIH Grant Application Writers Workbook?
No.
So I've used this for the NSF, but they have an NIH version of this, and I was curious if you had any opinions. So the NSF version of this, I guess I can show you guys it.
Well, I do see this first line. Good ideas submitted to the NIH are turned down more often because of poor packaging and presentation. That is definitely a very important and accurate statement.
So I really liked reading through at least the NSF version of it because it really reframed my thinking of not what you want to say, but what you need to tell people and how a reviewer thinks about grants much more than how a paper reviewer does.
Yep.
So I thought it was really interesting. It's like a stepwise, here's general format people do, you know, your overview of objections, then your specific aims. In specific aims, you have like potential problems, alternative approaches, like it suggests the sections and where they should go.
Yeah.
But I've had success with it.
I haven't looked at that, but I will say that, so I do often do grant writing workshops, and I have one PI who I've done them with a number of times. Oh, sorry. Man, that was annoying. And he talks about the things that he's learned over the years of trying to get grants, and he shows some of his early grant applications and summary statements so that you can see sort of what problems he ran into and how he's solved that. And one of the things that he talks about is that he has basically a template that he uses to write grant applications. And he, fills in the specifics every time, but he uses the same specific headers, sort of the same structure for everything. And it just helps him remember the important things he needs to put in there, the order that he has to put them in, etc. So operationalizing your grant writing, I think, is a good strategy.
Yeah, that's one of the things I really like about that book, because it outlines that for you. Like, start thinking about this one first, and then this, and then like take them on a journey with you, and it's always going to be very similar. What's the definition of early career? That was within 10 years of PhD?
That's a early stage investigator.
Okay.
So that's a very specific definition. And it's you haven't gotten a R01 or equivalent size grant, and you're within 10 years of your terminal research degree. So if you're an MD, it's different, but...
Oh, I see.
Okay.
So I gotta change to Dr. Mario. It's coming back?
Oh, yeah. I like this game a lot. I played this a lot.
All right, so we have two standard questions we ask on the channel. If chat gets slower, there's no questions. The one of my favorites is, if you had unlimited budget and no moral qualms, what experiment would you do? Or what would you fund? Say it that way.
You know what? I don't want to answer that.
That one's
because the moral qualms, I think, are really important, especially in genetics research. And so it's hard. Oh no. Okay, got it. So it's hard to answer that question. So I'm going to play the fifth on that. Sorry, everyone.
It's one of those like thinking about how far you could push something. Like Sarah's answer was, have a bunch of twins and separate them at birth. Obviously, that goes against IRB, but that would give you the most insights.
That would not go over well.
Exactly. Or another one was add an ankle bracelet on an entire town of people and send a hurricane and see where they go.
Okay.
So it's completely understandable you don't want to answer, particularly as a program officer. But we could say let's limit it to unlimited budget, but still moral qualms. What would you want to go after? What's the Manhattan Project of your area?
Well, I think some of the really important things are things that we're already trying to work on, and I don't know whether funding is. It's also just technology. Sorry. Oh my god. Okay. But so we've really gone down in the cost of sequencing substantially. But could we get that under $100? Could we get it to the point where your doctor can sequence your DNA while you're standing there in the doctor's office? Okay, I don't even know what I'm doing right now. What am I going to do with all these?
Ooh, horizontal.
Yellow. Oh, yeah. Oh,
nice.
Okay.
That's veteran.
I'm trying to get that there. It turned around and I didn't make it. So that would be great. And then we also at the same time have to educate physicians about how to interpret those results quickly and come up with ways to explain that in that sort of setting. But the faster we can do genetic analysis and the cheaper, the better.
So Jim Fadul wanted to sequence every male on Earth. Was his unlimited budget project.
Why?
I apparently from the male DNA you get I don't remember the exact rationale behind it, but it was basically if you had all those sequences plus tied it to every other piece of information we have, you could just data mine everything, like diseases, conditions, heights, weights.
I mean, a lot of large sequencing programs are giving us that sort of ability now. I just feel like I feel like I've gotten
Your
to.
life has fallen apart.
Yes. I'm really disappointed with myself here. But yes, the larger the data set, the more you can get from it. And data mining is very fun.
So CJ says, am I still eligible for that if this is presumably the early career review program? Am I still eligible for that if the research I do is not grant funded by the NIH?
Yeah, because actually, you should read the website. Okay, we just gotta. This is not going well. Yeah, gotta start over.
That's fair.
You should Google and look up that information. It's all there's an FAQ, I'm pretty sure, on the early career reviewer website. I actually think that you need to have an R01 or applied for an R01. There is some sort of requirement about that. We're gonna just, you know what I'm gonna do? No.
Just go up to 20 low speed.
I could increase the virus number and go up to a higher speed. It's harder when you're talking.
No, it absolutely is. People do not appreciate how difficult this is because you are being asked hard questions, like no moral qualms.
Yeah.
You gotta calculate that versus killing viruses.
Which is also important.
Exactly. All right, ladies and gentlemen, we're about an hour and a half in. For those of you just joining us, Ask a Scientist Gaming. Guest today is Dr. Lisa Chadwick. She is a program director at the NIH, does a lot with genomics and genetics, and is happy to answer any questions related to any of those topics, as well as others, plus some Dr. Mario.
So I'm failing at Dr. Mario at the moment.
Uncle Bill, welcome back. It's been a while. This is a fun one. This should become one of our standards. Reese's Pieces, rank the Mario characters by whose grant is most likely to get funded.
Well, I don't know.
So Scott Stagg got asked, which Mario character would you like to collaborate with on a paper? And his answer was Luigi.
Luigi. I'm really partial to, well, now I can't remember their names. Is it Toad?
Yep.
The little mushroom guys?
Hat, yep.
Yeah, I like them.
So Toad is most likely to get funded?
In Mario Kart, I haven't played Mario Kart for a while, but I feel like Toad was my go-to character in Mario Kart because the selection of vehicles was more nimble and...
Yep. Toad is one of the faster ones,
yeah.
I think. Yep.
This is... Oh, that's... Okay.
Acceleration, yeah.
If I was purchasing a sequencing... Oh, God.
So CJ wants to know, if your lab was purchasing a sequencing platform, what platform would you advocate for?
That is...
Oh, CJ, you're winning the night with questions.
CJ, email me later.
Yeah. No. Set up a call with Lisa. She'll be happy to chat.
I don't know. There are a lot of different platforms. They're all like pretty crazy to me, who I was originally sequencing using radioactivity and running them on a gel, and then developing the films and then reading it in nucleotide by nucleotide, routinely spilling radioactivity on my clothing, my shoes, and then you would have to put them in a drawer for like months. So the technology has really, really moved forward in ways that allow us to sequence. Hang on a second. Okay, much longer pieces of DNA and much faster. And I remember when I first started working at NHGRI, I really didn't know anything at all about the current sequencing technologies. And I saw people talking about what nanopore sequencing data looks like. It's basically like just Okay, we're just gonna let this go for a little while while I make hand motions.
You can pause.
It's basically just. Oh yeah, I can't pause, can I? Okay. Whew. It's basically just the DNA strand runs through like a pore, and I'm not going to explain this well. The electrical current like changes, and so what you see is just sort of this like trace of electrical current changes, and then the software can tell what nucleotide that means based on
Conductivity through the nucleotide. That's amazing.
Yeah.
No. That's really cool.
That's my technical description of that. I don't really know. That it just blows my mind what we can do with it now. But I really did Sanger sequencing, and then after I did it with radioactivity, I did it on an ABI sequencer for a long time. So I'm just dodging your question, CJ, but thank you. Thanks for asking.
Kudos. Thank you for the question. That's amazing. All right, I got to figure out which other ones I want to ask. So one of the things we have as scientists is we regularly, you're getting on a flight soon, a long international flight. And there's a regular airplane conversation. So when someone asks what I do and I say a chemist, their natural response is, I hated that class, or chemistry, that's hard. So if Sarah does the same thing, she's a psychologist, so they want to ask about schizophrenia. If Jenny Root does, she studies like developmental disabilities. People want to talk about the person they know that has disabilities. So what is your airplane conversation when you tell people what you do? What do they follow up with?
I usually just say that I'm a scientist, and even they don't need to know that I haven't worked in a lab. Like, I'm still a scientist.
Yeah.
And maybe I'll say that I do genetics research, or that I'm involved in genetics research. I actually have not. Well, it's been so long since
post-COVID.
Since I've been in a plane.
Yeah.
The institute I used to work at, one of my portfolios was the microbiome. That is something that people are always interested in talking about. So that would always generate some interesting conversations, like how can I change my microbiome to manage my weight better, or like people, or to like fix some sort of thing that they think is wrong with them. So I would say the microbiome is one of
the microbiome. What can I eat to solve my problem?
Yeah.
What gut bugs can I have?
Yep.
CJ, do you tell people you work at the NIH? Pre-COVID, I would mention NIH. Most people didn't know what that was.
It's definitely changed in relevance. And then the next question that everybody asks me is, do I know Tony Fauci? No, I do not. I wish I did. He seems like a really great guy.
I mean, good on him for getting rock star status in the science world. Like, not a lot of people get that, so.
He may not have wanted that.
Yeah.
But yes, so I do tell people that, and you're right. More recently, people know what that means.
CJ, I saw him a couple times going into Building 31.
Oh,
Yeah, and it's just paparazzi taking photos of.
He came and gave a talk at our NHGRI symposium one year, which is like an institute-wide event that we have every year. And it was a really interesting talk. It was just basically about, how this was pre-COVID, but he talked about sort of the role that he's played in different pandemic responses over the years, and like how he's interacted with different administrations and some of the challenges, and it was just really interesting. I used to see Francis Collins sometimes in weird places. He used to be the NIH director, and now he is back in his intramural research lab at NHGRI. But I remember seeing him in the lobby of a hotel just walk by with his guitar. I was like, wait, is that Francis Collins? What is he doing in this hotel?
Oh, man.
With a guitar, but he likes to play his guitar in places.
We need to Ask a Scientist music. Ask a Scientist musician?
Yeah.
Ask a Scientist jams.
Yes. There are a lot of musician scientists, myself included.
Yeah. Ask a Scientist unplugged. Feel that rough?
Oh.
No worries. That's pretty fun. We should do another prediction because we have a bunch and we're running out of time. It's already 9:45.
That's hard to believe, but
That's
okay.
not crazy. Time flies.
Let's do that one.
All right, ladies and gentlemen, it is time for another prediction. If you're not following us, click the follow button. One, because that gives you additional internet points, but two, because it increases our visibility. So please do that now. We're going to pop up a prediction. This one's an interesting one because there's no possible way you can know the answer to this other than extrapolating from what you've experienced for the last hour and 45 minutes. So the question is, Lisa has eaten giant grubs, yes or no? Has Lisa eaten a giant grub? So I did a tequila tasting. So my wife and I celebrated our 40th birthday and we went to New York City and just like without the kids and had fun. And we did a tequila tasting, which part of one of the tequila shots was eating like fried grasshoppers beforehand.
Oh.
Oh, Almost PhD done has redeemed take a drink. So
Okay,
cheers,
I'm
Lisa.
pausing it.
Absolutely.
Cheers.
Thank you. Almost PhD done. Congratulations on almost having your PhD done. Cheers to you as well. Thank you for making us drink on a Wednesday night.
I hope next time I'm on this stream, your username is now PhD done.
Exactly. PhD completed. All right, Lisa has eaten giant grubs. We're at a 50-50 split, ladies and gentlemen. Bet your standard internet unit's useless anywhere else except for making us drink alcohol and maybe requesting a factoid. It's totally up to you. But yeah, almost PhD
Pretty
done. Thank you for the
stressful.
follow. We didn't get a follow notification, did we? But yeah, thank you for the follow. Thank you for joining us. If you have any more questions, throw those in chat. I promise we'll get to them. If I forget, just remind me, because we get distracted by both gaming and talking about random stuff. So we're going to try to answer all your questions.
I really need some red ones here, Dr. Mario.
Now, this is so in the gaming community, we call it RNG, which is random number generator.
Yes.
Just shits on you with all the wrong things.
Just one after another.
There you go.
Okay.
Nice. All right, you have about 15 seconds. Put your predictions in there right now. Lisa has eaten giant grubs, yes or no? What have you learned about her playing Dr. Mario that relates to eating grubs? We'll find out. All right, so Lisa, what is the answer? It is a 50-50 split in terms of betting.
Wow. The answer is no, I have not. But I did just go on a trip to Ecuador, and I watched other people on our trip eat giant grubs. I am a vegetarian, and I decided, whether or not this is reality, that grubs are not vegetarian, so I did not eat any.
That's interesting. So there's a I don't know, deciding a threshold of what to eat. There's like this level of consciousness or awareness threshold that people have. Like plants are alive, but they don't have a circulatory system, they don't think. And your line is drawn below grubs is what we learned.
Absolutely.
Yeah. That is fair. So you watched other people eat grubs?
I did.
Were you curious? You
Nope.
were tempted?
Now,
Not even a little bit?
my son, who's 15, was on this trip with me. This was a 2020 planned school trip to Ecuador, which we didn't take until this year. And he eats meat, and he likes to try different meats. We call, so like many kids, his first meats were chicken, like chicken fingers. And then
nuggets,
he became interested in eating different kinds of meats, and so we would call those advanced meats. So I tried to get him to try the grub, and he was like, I think that's too advanced for me.
Oh, man, it's a life experience, though.
So he didn't.
Oh.
But one of our friends who I thought might join this stream to watch, maybe he'll be here at some point, he did eat them.
And his feedback was?
I mean, some of them ate more than one, so I don't know how to feel about
they might
that.
be delicious. I've never had a grub. I don't know.
Okay.
That's pretty fun.
This is a lot easier to play on slow speed.
Yeah. And the speedruns are amazing because they just drop them down.
I mean, if I was really concentrating, I could do this a lot faster than this.
So this question, like I do a lot of test analysis. I teach general chemistry, so I have like n equals 350 data points every time I ask a question. This one's intriguing because this is one of those like, why would she ask this unless the answer was yes kind of questions?
Mm-hmm.
But you threw a twist in there.
Yep.
And so congratulations to those of you that said no. In fact, one individual said no. GREGoR, we're going to say that's how it's pronounced. You get a 2-to-1 payout, so congratulations on your confident bet.
Good job.
No. I'm curious what led them to believe no or yes? Or betting against the stream? I like it. So Dr. Mario was the underappreciated dropping things in a pile game behind Tetris. Like everyone and their mother played Tetris, but Dr. Mario, I feel, was really underappreciated in this genre.
Yeah, I think I played I've played everything. Mm-hmm. So I definitely played Tetris a lot on my Game Boy. Mm-hmm. But I played a lot of Dr. Mario in college. I had my not a Super Nintendo, a regular Nintendo in my dorm room, and I played this then. I played it with my brothers. I basically have internalized the music for this game.
It does have some jams.
Well, it does. You can choose Fever, Chill, or you can choose the music. So I have not switched it from Fever, but no. Where am I gonna put that?
Fever versus Chill, that's right.
Yeah,
and they're accurate descriptions of the tone of music.
As soon as I hear the other music, I will be like jamming out.
Yeah. It is quite chill.
I think I was hoping that you were going to play these games too.
Oh, sorry, I didn't let you know. No, I'm a tourist. I just watched the chat. Oh, do you want to play versus mode? We could set it up. It's up to you. All right.
Okay.
I'll get a controller. This is one of the games I will play.
All right, everyone. Get ready to watch me get destroyed.
We don't have to be on the same one.
What fun is that?
I'm going to have to start it, restart it.
All right.
Ready? Mm-hmm. All right. Ladies and gentlemen, if you have questions, throw them in chat. I'll get back to them after we play a little bit of Dr. Mario. All right. Let's do it.
Okay. Should we? Oh, let's do music chill.
That's important. Let's do low speed.
Low speed. Okay. And should we start at 10?
All right.
Oh, wait, I have to get to low speed too.
Oh, man. I don't think I've played 2-player on stream. This is a first.
Am I on the
You're
left?
on the left, probably.
Okay, good, yeah. Oh, I forgot that was going to happen.
Oh, yeah, no, you can shit on each other.
No. Now I have to move faster.
Yes. No, now it's stressful.
I've made a terrible mistake by asking you to play this with me. But I was just gonna...
Oh, man. No, my wife and I used to play this all the time together. Pre-kids. Oh, God, that's gonna cost him. Sorry, everyone. I'm completely ignoring chat while I play some Dr. Mario.
I know.
This is nostalgia. We're going back to childhood here.
Ooh. Oh, you're really like... Okay.
Yeah, I played this a lot. This was my go-to strategy game. Ooh, diagonals.
I need to like move a little faster.
Yep, you gotta drop them for sure.
I'm not moving fast enough here. God, you're killing me. This is not going well for me. Really regretting my choices.
But I'm happy you asked me to play. Because I love doing Ask a Scientist gaming, but most of my spare time now is setting up games for other people. It just really cut into my game playing time.
It's really I need to get that. Oh,
so I will take this opportunity.
Sorry, everyone whose ears I just yelled at.
Sorry, headphone wearers.
I was trying to get that.
All right. Oh, okay. Drink pot. Okay. All right. Anyone just joining us, Ask a Scientist Gaming, the guest today is Dr. Lisa Chadwick. She's happy to talk about genetics, genomics, the NIH, any questions you guys have. We can even make up answers for other fields. If you want to ask astrophysics questions, feel free to throw them in chat. We will address them shortly. Right now, we're playing a little bit of Dr. Mario, just for fun, special occasion. I'm actually playing video games on stream with my guest.
He's talking really calmly while he's like completely destroying me, and I am over here not even trying to make conversation. And
I mean, to be fair, I play a lot of video games. It's really fun as I've started playing them with my daughter. I don't know if you've heard of the book series Last Kids on Earth.
I haven't.
But it's about a zombie apocalypse where there's just a group of like teenagers that survived. But there's a video game associated with it where you go around killing zombies, which is pretty fun.
God, my system has broken down here. And I don't know what I'm doing.
Oh yeah, when you get the mess on it. Worst part is it takes a lot of time when that stuff drops. What's crazy to me is if you watch Twitch streams, like some of the best players in the world will play this at like epic levels of professional gameplay, and they're just sitting there reading chat and chatting simultaneously, and I don't know how they do it.
I can't do that.
I mean, did any of your 1,000 hours of Zoom meetings prepare you for this moment?
Why did I do that? Why did I do that? No. Now that's gonna be hard to recover from. I did used to play this with my brothers. I have 2 brothers. But other than that, it's been a long time since I've played 2-player. It's been a long time since I've played any player Dr. Mario.
Yeah.
Especially 2-player Dr. Mario.
That's fair. I'm making a mess. It's fine. We'll recover.
Maybe.
Let's be optimistic. That's a good one.
Oh, come on. Now that just over there, like, that's...
Oh, come on. You could have had that. Unacceptable. All right.
Oh, jeez. Oh, my God. I was trying to do something and I missed. I'm very competitive, too.
But you knew I speedrun this game. I don't know why you chose this.
I know.
I used to be top 50 in speedrunning this.
As I said, this wasn't my best life choice. Look at this. All right. Well.
Cheers.
Good job.
Oh, we got one more.
Oh,
we got 3 crowns to win. Yeah. For some reason, it's not best 2 out of 3. It's best out of 7.
Okay.
All right, we got this. Anyone throw your questions in chat. We're focusing on Dr. Mario right now. We're happy to talk science.
That needs a home. Oh, that was the right home.
Oh, God, that's going to hurt him. Can he come back from that mistake? Yes, probably. That's okay. I think one of the keys to this game is just realizing when you have to sacrifice, like make a mess.
Yeah.
Just accepting that reality.
Well, at least those fell on the right colors for me. See, that was a great plan that I had over there that just got totally messed up. Oh my gosh. Okay. I think that
just a relaxing Wednesday night, drinking, playing video games.
the biggest question that I have is, how am I supposed to go to sleep tonight after I have been stressed out playing? Okay, this is...
I mean, the key is download this for your Switch.
I know, actually, I don't have this one, so...
So it has an NES emulator on the Switch. So you could play this if you wanted to.
Okay.
Anyway, cheers, thank you. I haven't played Dr. Mario in like a year, so that was fun.
Okay. What else should we play next?
So we could do this again if I go a high level and you go a lower level. We can make this competitive.
All right.
Let's do it.
Fine. Wow. Okay. Ooh. Okay.
Yep. No, we're piling it on. See, if you're gonna beat me, it's gonna happen very early. Oh man, TJ requested a factoid.
Okay, TJ.
You need an additional handicap because it's coming. That's awesome, CJ. Thank you. That's almost cruel.
Oh, okay.
You can pause
So,
if you want to. Nope.
I'm on it.
All right.
If you put all DNA in one cell, end to end, it would stretch 6 feet. Fun fact about DNA.
So say that again, all DNA end to end.
Yeah, all the DNA in one single cell. If you like stretched it all out, unwound it, it would be about
that's
6
from a
feet
single
long.
cell. Because I remember Scott Stagg had a similar factoid, but it was all the DNA in the human body, and it goes to like Pluto or something.
Oh wow.
Yeah.
Okay,
I've learned a lot on these streams. Like I'm an expert in photochemistry, but just factoids and questions and.
I'm actually talking about how you're making such good progress against me in Dr. Mario, but yes, that too.
Is there a limit to the number of drinks that can be taken in a row? No, we have not put a limit on that. So you can make us get hammered.
I didn't drive. So.
Oh god, there's a whole bunch building up right now. They're messing with us at this point. So we should probably pause and take a few drinks.
All right.
I gotta get a new
I
one.
actually need to fill my...
Grabbing another just to make sure you have one.
Okay.
All right, cheers. So we have CJ redeemed. Take a drink. CJ redeemed. Take a drink. Cheers, Lisa. CJ, thank you for joining us for some Dr. Mario and drinking. Drosso Killer also redeemed. Take a drink.
All right.
CJ, holy shit. Oh, factoid. Take a drink. let's just chug a bunch. CJ, make it rain. Speaking of which, we should do a prediction because it's 10 o'clock already, and we're.
Yeah, I definitely want to give you guys more
All right.
ammunition to.
Which one?
Okay, that one.
All right, this is a fun one. Back to the science questions again. If you're not following, click the follow button. Use your standard internet units to make us drink alcohol or request factoids. Also, you can bet on questions and decide whether. Give me a second. All right, so the question is, what is the speed record for genetic disease diagnosis? And so you guys have 2 minutes. Is the answer 7 days or 7 hours? And so what is this speed record start to finish? This is from
Like
initial recognition to full diagnosis?
Yes. Initially seeing a patient to diagnosing them. CJ, you better get this right.
7 hours or 7 days? Yeah.
Let's play while we're waiting. Just wanted to drop that on you
That's fine.
while you're unaware.
Oof.
It's really like embarrassing to me though how well you're doing, given how differently we started this.
Oh God, he is not doing well now. I am sacrificing. Alright, there we go. Get to clean up a little bit. No pressure. CJ, just making it rain with your standard internet units. I respect that. You earned every ounce of that. Making it rain.
CJ is probably going to win some more in this prediction.
Oh, we'll see. All right, you guys have about 20 seconds left. The question is, what's the speed record for a genetic disease diagnosis? Is it 7 days or 7 hours? I'm looking across the screens. That was a mistake. I regret doing all of that.
I don't. I might actually win a game.
I gotta tear down some of this wall.
Yeah, you've got problems.
I do. I have a mess.
Oh, you're not making things great for me, and I just made a dumb mistake.
All right, so Lisa, what's the answer? 7 hours or 7 days?
It's 7 hours, and I believe 18 minutes, actually.
It's down to the minutes. That's crazy.
It is
trying to
crazy.
get anything done in that period of time is insane.
It is crazy. It just happened, well, it was just published this year. Some of the researchers that were involved in that, well, it was a group from Stanford. Some of them are part of the GREGoR Consortium.
Mm-hmm.
And it is really a big deal because you, especially in like the NICU, you see a lot of like critically ill babies, and you need to know what is wrong with them as quickly as you can. And I think I said earlier in the stream how long, it used to take to understand or to find the gene that was causing a genetic disease. Years and years and years and years and years. And that's years that those people are waiting to understand what's wrong with them, to think about what kinds of treatments are the right treatments. I'd like to hear you make something like that. That means that, although I've got a little bit of a mess here right now, but I think I can take care of it. Oh, well, you've just screwed that up. So, thanks a lot.
Spoke
too soon. So this enables us to get to an answer a lot faster, which is a huge thing. People who have rare genetic diseases are often on what we call the diagnosis odyssey, where they're just for years going from doctor to doctor to doctor, trying to figure out what's wrong with them. Oh my, I cannot believe that I am going to...
Oh.
This is not...
What kind of sequencing did that involve? Is the question.
No, I actually think that was... I don't want to say incorrectly. But what I do know is that what they did was like run many times in parallel. So we talked earlier about coverage. And they ran it like the same individual like in 42 different wells on a sequencing run, so that they could get to that high level of coverage quickly. That was one of the biggest innovations, I think, that in that effort. So but I'm sorry, I don't remember whether they did exome sequencing or genome sequencing. Probably exome sequencing. It is like, because I'm pretty sure it was probably exome sequencing, but you should read the paper.
I mean, so one of the
in the Lancet.
inherent problems with a rare disease is in a capitalistic market, there's no incentive to cure this stuff, right?
Yes.
Like, so how does... I mean, obviously the NIH funds some of that effort, but how does one deal with that cost benefit analysis.
Yeah. So I will be honest that part of it is a little bit outside of my area, but there are other parts of the NIH that, oh, why did I do that? That are really interested in that sort of development of treatments for disease, all diseases. But there is a consortium that,
As long as you're having fun.
there is a research consortium called, I hate this acronym, I hate saying it, IRDIRC. It's International Rare Disease Research Consortium.
IRDIRC.
Yeah.
We'll see if Twitch flags that one or not.
I know. So that consortium is made up of why am I doing that? Made up of funding agency representatives, researchers, and companies. And a lot of what they talk about in that consortium is actually that sort of more patient-centered aspects of rare disease. So how can we make it more affordable for people? How can we reduce the length of the diagnostic odyssey? How can we incentivize companies to develop treatments for rare diseases when there's only 60 people in the world who have that disease? How do we get those treatments through clinical trials? Like when you...
So that's expensive, right? The clinical trial process is not cheap.
Well, it is, and you also need to have a certain number of people involved in it.
I see. And
when it's a rare disease, you just don't have... No, that... I had a way I could have dealt with that, and I didn't, and now I'm paying for it. So there are different sorts of ways of doing trials that they're talking about, and I don't want to talk in too much detail about it because it's a little bit outside of my area. But there are people thinking about ways to solve that problem, but it is definitely an issue.
I mean, and that sucks. It's not an unreasonable issue that somebody has to consider. It's
right.
Like cost benefit, actuary tables.
That's true.
I'm trying to look over a chat, but it's speeding up.
I'm just gonna sit here.
That's really impressive, given that I assume about an hour for DNA extraction and then at least a couple hours for sequencing itself. Yeah, that is amazing.
Yeah, it was really a major thing. They got a Guinness World Record for it.
That's awesome. Claims to pay fame. Like H-index, sure, but if you have a Guinness World Record...
how many scientists can say that?
Like that goes in your CV.
Okay, what am I doing?
So how do you feel? So the NIH has are you familiar with the broader impacts section that the NIH now requires? It's a recent development within the last, I guess, decade or so.
What do you...
So it's a very particular section about you have to explain.
Oh, NSF.
Yeah, NSF, not NIH.
Yeah, I was like, no, So I have heard about that, but I don't know a ton about it.
Yeah.
We sometimes have grant announcements that we do with NSF, and I just heard about one recently.
Oh. And so that's you have to.
And so I heard some discussion about that. I think that's really important, but tell me what exactly...
I mean, so it's a bit nebulous, but you have to basically formalize how this is going to impact society. And so that's a combination of what's the implication of your work, but also typically some kind of science outreach endeavor.
Yeah.
So this is technically part of my NSF grant, doing this Ask a Scientist stuff.
Really?
Yep. So every annual report I write, here's how many guests I had, here's how many viewers we had. And so, but it's also really annoying because it's the most nebulous part. Like, how do you weigh this versus someone going to a high school classroom and talking about science kind of thing? So
that's interesting. We do that sometimes in some of our funding announcements. We have a program at NHGRI called SEGS, which stands for Centers of Excellence in Genomic Science. And in the SEGS, there's an outreach
there's
component.
a formalized.
And a lot of times what we're talking about is like outreach to other scientists. Like you develop a technology and we've paid for you to develop that, but how do we get you to get people to use it? Okay, like I have a real problem over on the side there. Oh, you've got a problem as well.
Yeah.
Okay.
No, you got to build up from the bottom.
All right. So it is really important because we're taxpayer funded.
Yeah.
And it's important that taxpayers
service.
Understand what we're doing. And you see all the time what happens when we don't do a good job of explaining to lay people what we're doing, why we're doing it, why it's important. I can't remember which politician this is, and I don't want to get too...
Oh, is this fruit fly related?
Well,
Sarah Palin.
No.
You talk shit about fruit fly research. Why are they funding it? Yeah.
Yeah, so there's one guy who would go through all of these grants and then would publish like, here are the most ridiculous things that I saw.
Yeah.
And that's, we know that basic science is really important. But the lay community, if we don't do a good job of explaining to people why it's important to study how Drosophila do X, Y, and Z, then they're just like, why are they putting flies on a treadmill?
Yeah,
I don't think anyone's actually putting flies on a treadmill, but it is really our job to do a good job of outreach like that.
Yeah, the reason I mentioned Sarah Palin is that was in the, I don't even remember what election that was, but she brought that up as being a big deal because it was like, fruit fly research, why are we studying flies? And that's like the foundation of modern genetics research, and it's just really underappreciated how impactful that actually is. Yeah,
and how much of what we know that has enabled us to, sorry everyone who I'm yelling in their ear. I'm so sorry.
You can pare back by asking a question right now.
But to get to the point where we're developing treatments for things, that only happens because of all that foundational work that we've done. So like getting the COVID vaccines as quickly as we did, that's because all of this foundational work has been done for years and years and years that enabled us to be able to quickly turn it into something that benefited us.
Yeah, something useful.
And people don't appreciate that.
No, absolutely. My favorite example of that is Einstein's relativity and how GPS couldn't exist without it.
Oh.
Like, you can't speculate how this foundational research will be useful.
I've got some problems here.
It's okay.
I don't even know what I'm doing with this here. Like, just trying to turn that around. Okay. Wow.
Right. Yeah, the fruit fly geneticists came up with all the dumb gene names.
I would not say dumb. I'm sorry. I'm actually really jealous of all of the gene names.
Wait, what are the gene names? What are the absurd
Actually,
ones?
there is a gene called Sonic Hedgehog. So Drosophila geneticists got to name the genes after the phenotype that they caused, and they came up with all sorts of great ones.
They're all just acronyms of fly phenotypes. That's fair.
Yep.
Go Haku. Why is science looked down on by politicians?
I don't know.
It's not universally looked down by politicians. There's a few very vocal ones that are going to complain and try to cut the NSF and DOE budget. But for the most part, I think science is pretty well supported, right?
It is. I mean, it is one of those things that has bipartisan support every time. As far as I'm concerned, we never have enough funding for scientific research. But,
I agree.
Yes, I think in the chat you should continue to say fly gene names because there are a lot of great.
Son of the Seven list. That's a fly gene name. That's amazing.
Yep. Okay.
You want to keep going or something else? Because we have a little bit of Top Gun, some Castlevania.
Oh, yeah.
You want to do Top Gun?
Sure.
You have about 10 minutes until we could bust out NARC.
Okay.
This was a last-minute edition by Lisa.
So, I told my brother I was going to do this, and he said, oh, well, you need to try Top Gun and landing on the aircraft carrier, which I had totally blocked out how many times I tried to do this when I was younger. And it's not easy, and this is not going to go well.
No, it's not. This is terrible. Because
I
there's
haven't
no
played
instructions.
this game probably since I was in middle school.
There's no instructions or advice how to land on this aircraft carrier, and it's where basically everyone stopped playing.
Yeah.
Iceman. I haven't seen the new Top Gun movie. I apparently have to.
So they stick like onto protein names. So actually, the first gene that I cloned was BHMTL, which was BHMT-like. BHMT is beta homocysteine methyltransferase. I don't even know what I'm doing here. So okay.
Pick the T11 rocket.
Okay.
Are you
All
ready
right.
for this?
No. Take off. I don't even know how to do that. Oh, I guess I just took
There you
off.
go. I'm gonna run to the restroom while you battle this.
Great. Great. Hi, everyone. I don't know how to shoot or... Oh, okay. This is looking sort of familiar to me. So someone's behind me. How do I shoot them? Oh, jeez. I feel like there's a way to lock on to them. I don't know what that is. This is going well. Hope you're all enjoying watching me kill no one in Top Gun. Not a single... Oh! Got one. You're all too far away. No, 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 no. Oh. No. Okay. This is riveting, I'm sure. Thanks everyone for being here. I have killed one plane, a total of one. I've basically just flown around and then I got shot.
It's more than zero. We'll take it.
I thought that there was a way to like lock on them, but
So one is bullets and
danger,
one is rockets,
danger,
right?
danger. Oh my God, what danger? Oh, somebody's behind me. Okay.
So have you had to deal with politicians at all? Obviously NIH has people that talk directly and lobby and...
I don't know. I do help prepare information for people who have to talk to politicians, but I don't. We do have a whole office of people, and if you're interested in... Oh, I'm not even going to get to the aircraft carrier. If you're interested in jobs at the NIH, which is a fantastic place to work, I love it, and I highly recommend looking into it. There's all kinds of different jobs that you can have as a scientist, but we do have people whose job it is to specifically do like congressional liaison kind of stuff. And I don't know that they necessarily are lobbying, but what they're doing is understanding what I'm doing a lot better.
Shredded.
They're understanding what Congress is asking of us and how we can sort of fulfill that.
So if you don't mind me asking, salary for a program director? Like I know at the NSF, I think starting is something like 140,000 per program. Yeah, but they're living in DC, so I don't think they can work remotely.
So the nice thing about okay, here we go.
Did you watch the YouTube video I sent?
No.
Where's your ASCII art now?
So everyone, get ready to watch me crash a plane.
We're gonna landing sequence. You have to control your speed and your elevation to something. It's like your speed has to be 200.
How do I do my speed down?
I think it's the up and down, right? So if you go up, it slows you down.
Oh. speed down.
So you have to do like altitude. I don't remember the numbers. But there was no instructions on how to do this when you first got the game.
Left,
So it was just
left.
like randomly try shit.
Speed up now. Okay. Oh my god, I'm gonna crash.
NIH is Title 42 pay schedule, so they are their own thing.
No. Program officers are not Title 42. We're on the GS.
Is that a postdoc?
Yeah, I'll
related?
explain it as soon as I've crashed this
like your
plane. I'll
focus won't
explain it.
matter at all.
Wait, I feel like I'm not going fast enough. Like I'm gonna
No, you're gonna brighten the drink underneath.
speed up. I don't know how to speed up.
I think it's nose down, speeds up. It's like that is the literal full Top Gun game that I remember playing. Like you shot the stuff and then you crashed a plane.
I'm not playing that again. Let me answer this question.
Yep.
So there are some positions at NIH that are Title 42, but most of positions like mine are on the GS. The way that I think actually the state is very similar to this, that there's like very specific pay grades. And then within a pay grade, there's like different steps of salary. And you can see what the salary tables are for that. It does depend on where you live. So I am a remote employee. I live in Tallahassee, and I have for a long time. So I don't get paid the same as my colleagues at the same grade and step as me who live in Bethesda. I get paid at the Florida rate. I'm not playing that again.
All right, what do you want to do?
I don't care.
So we have about 5 minutes till NARC, unless you want to start NARC now. It's up to you.
Let's do Dr. Mario for 5 more minutes.
Okay. Or Castlevania.
Oh, we didn't even do
No, we didn't. Do you want to do that?
Sure.
Little bit of Castlevania.
Okay. So anyway, program officers are anywhere from GS 12 to GS 15. So you can look that up. GS pay scales. All right, this is a game I also played a lot.
Bethesda has a locality of 30%.
Yeah, it is.
What does that mean?
So there's a base salary for every grade and step level. And then depending on where you live, they've negotiated increases to that base rate. So it's not just Bethesda and the DC area. It's like any like major metropolitan area. So Florida is, well, at least in Tallahassee, we're in the rest of US, which is the lowest. And since this is where I live and this is my duty station, I get paid at this rate.
Yeah, and that makes sense. And to some degree, it's fair. We can actually buy houses here, so.
Yeah, right.
Yeah.
Exactly.
Yeah. All right. Huggy Bear wants to, or says, I saw a CRISPR at home kit. I'm excited for the opportunity to learn more about it, but I was worried what people might use it for.
Yeah, me too. I saw that too. I'm not sure I would recommend doing that.
I mean, so have you seen the glowfish that they sell at like PetSmart and stuff? The UV lamp, the GFP functionalized?
Yeah.
And how long before somebody wants a glowing infant?
Well, I mean, hopefully a while. But we've been using GFP in research for a long time. Actually, I think when I was in graduate school, we thought a great business plan would be to make tattoos out of GFP. So that your day-to-day, you would have no tattoos. And then when you went out to like a dancing place and they were like black
UV lights.
Yeah. So
I mean, those exist now, the UV tattoos. I don't know if you've seen those.
What?
Yeah. Ultraviolet tattoos. They're fluorescent. They're inorganic fluorescence, not like protein based, because they last much longer.
Why didn't I get a patent on it?
No, exactly. No, they're.
I would be rich right now.
I mean, it's.
Oh,
right in the drink.
I used to play this game. I didn't have this game actually, I don't think, but I babysat someone who did. When I was in middle
so
school.
there was a researcher in China. He CRISPR'd out HIV or something, right? He got arrested and was recently released. But there's kind of a genetic free-for-all because the rules aren't clearly established yet, as far as I know. Or are they?
No, they're not. There are a lot of things that you could do at home, I guess, if you were really motivated, and that doesn't mean that you should do them at home. When I had microbiome in my portfolio, I mentioned earlier that that's something that people are very interested in, figuring out how they can manipulate. And I remember being at a meeting once and walking to lunch. NIH people, when we go to meetings, we have to buy our own lunch.
Oh yeah, your rules are pretty crazy.
Yeah, they are. So I was walking to lunch and I saw this person, and I assumed that since he was also walking to lunch, he must be at the NIH because the meeting had lunch. And so I started talking to him, and he was like, "Oh no, I'm at this meeting because I'm interested in doing home fecal microbiome transfers." And I was like,
injections.
Really? Yes. And so I understand, like, people who have, like, let's say you've got Crohn's disease, your quality of life can be really significantly affected, and you are desperate to find a solution for it. And you'll try anything. The thing is that you don't always know, for example, in that sort of, I have to kill this guy,
first boss fight, yeah.
Yeah. Okay, I can't just, like, go into a door and get rid of him. You don't know what you're aiming... Thanks a lot, bat guy. You don't know what you're aiming for. So just because you can manipulate your microbiome by transplanting someone's poop into your intestines, how do you know that that person's microbiome is the right one? How do you know that that's, like, the state that you're supposed to be trying to get to? I don't even know what I'm supposed to be doing. That bat is gone.
I thought you were supposed to kill that bat.
But he's gone, and I can't go anywhere.
Go Haku, you poop transplant.
Yeah, I mean,
but there's been legit research studies that show this works.
So you might have heard of C. difficile infections. That's like when you go into a hospital, you're on antibiotics. It kills your normal microbiome, and then you get an overgrowth of a bacteria called C. difficile, which is normally in your intestine anyway, but it's sort of kept in check. Then the best way to solve that is through a fecal microbiome transplant. Could lactose intolerance be solved with someone else's microbiome? I mean, so lactose intolerance, I know, does have a genetic component. I don't know a lot about the specific genetics of it, but yes, how your microbes help you digest different foods, different parts of food that you don't necessarily know how to digest yourself. And so maybe it would help.
So lactose intolerance, that's not an allergy, that's a digestion issue?
Yeah.
That's interesting.
That's because you don't produce enough lactase.
All right, we're at 10:30. We should do a prediction, and then we should flip over to NARC.
Okay. Since we just played Top Gun, let's do that.
A Top Gun related question. Ladies and gentlemen, if you're not following us, click the follow button, get your standard internet units. It is time to gamble on a question. This is a Lisa related question.
I legitimately do not know what to do right now.
I think you're soft locked. I think something happened that it glitched out.
Yeah, I think so too.
That's crazy. If it would have made it faster, that would have been a speedrun strategy. All right. Lisa has a pilot's license. True or false? What did you learn about her Top Gun gameplay that led you insights into whether she has a pilot's license or not? We'll see what the answer is. But yeah, click the follow button, use your 300 standard internet units. You can still make us drink alcohol, although we've had quite a bit already.
Yep.
But now it's time to play NARC. For those of you new to the channel, NARC is the game we close every single stream with at 10:30. We're going to speed run our guests versus each other on how fast they can beat this game and win the war on drugs. Lisa, are you ready to just say no to drugs?
I mean, I'm always ready for that.
All right, press start. Let's go. What medical certification do you have?
I'm not answering that question.
I
know.
But what pilot's license does she have? All right. So here's your timer if you want to see it.
Oh.
Press start button.
Oh.
There you go. All right, this is NARC. You're gonna take on the drug dealers. Walk right, find doors.
Oh, do I need that?
No.
Oh.
You don't need anything. You just need to go right. It's a relevant question, though underhanded.
It is.
I mean, that's one thing we have to deal with on stream in terms of what you actually want to talk about and what you want recorded and publicly shared versus YouTube. Because off the record, I would love to hear.
Once we answer this question, I know why.
That's fair. All right, you have about 15 seconds. Does Lisa have a pilot's license? Yes or no? True or false? Does she know how to fly a plane? Not in NES space, but maybe in real world, does she have a pilot's license? That's a fun question. I like the mix of like science and personal because it just, again, the humanizing the persona.
Gotta get through this.
All right, Lisa, what is the answer? Do you have a pilot's license?
I don't. I have flown a plane before. My dad has a pilot's license.
Flown a plane means he let you take the controls?
Yeah. There are two sets of controls in a plane. So I have flown the plane. I actually don't think I flew in a commercial airliner until I was in high school on an orchestra trip.
Oh, that's fun.
Because we used to fly in my dad's plane everywhere.
Oh, like he had a private plane?
Yeah.
That's awesome.
So
I mean, but also expensive and maintenance.
It is
like pain in the ass, I imagine.
It is expensive.
Yeah.
Gas is expensive.
Yeah.
But yeah, we flew to. I grew up in Wisconsin.
Oh, did you?
Yeah.
That's awesome. I'm from Minnesota, actually.
Oh, are you?
Yeah, St. Cloud.
Oh. Okay.
So I'm from backwoods Minnesota.
Yes, you are.
I'm Michelle Bachman's district.
Wait, why can't I get through here? It's because I need to.
So you need to, you need to
some
bullet
stuff.
the guys in the black shirts, but reasonably close so they drop a key.
Jerk. I'm.
Tonight we learned that Lisa takes games seriously.
I really do. I do. It's a problem.
It's not a problem.
No.
That's my problem. So
I want to win this.
So believe it or not, I used to be the world record holder in this speed run, and someone beat me 3 days ago. So I am as of 3 days ago. Yeah. So he beat me by 3 seconds. It's like, do I want to lose sleep to get that record back?
If it was me, answer yes.
So the answer is I feel for you. I understand the strain associated with this. In fact, right now I'm editing a speedrun tutorial on how to beat this game as fast as possible.
I mean, my left thumb is really hurting right now. Do I have to get in that car?
Yes, you do. So jump button to get in the car.
Wait, what's jump?
Tap B.
Got it.
Tap B when you're a little lower.
He just killed me, not that low.
Yep, there you go.
Oh, I was in it. Oh. Okay, wait.
So the problem is you're going to run into either a wall or mines, and there's nothing you can do about it unless you watch my speedrun tutorial.
Oh, God. Okay. Whoa! I wasn't expecting that. Okay.
Exactly.
What was the point of having a car at all?
Unless you memorize this path, there's just no way you can possibly do that. So this is a very unfair game. All
right.
I mean, to be fair, most Nintendo games are super unfair.
At least I'm walking and no one's shooting.
All right, we're closing the night with NARC. If anyone has questions for my guest, Dr. Lisa Chadwick, questions about NIH, questions about genetics, genomics, please feel free to throw those in chat. We will discuss them whether we know the answer or not. We are happy to do so. Any opinions on the Allen Webb telescope images? Have you seen any of those?
I have. How do I get over this? Oh, I just jumped. Okay, fine. Thanks. It is really impressive.
It's one of those I talked to, who did I have on? I don't remember who it was. It might have been one of the particle accelerator people, but like the US used to invest a lot in like big science. You know, like the Manhattan Project or like these particle accelerators. And we've kind of teetered off on doing that. You still have like Allen Webb Project, but like new big science endeavors just don't happen as much now.
Yeah, and part of that is, I think, the tension on these dogs. Like,
oh, you can, if you hold both buttons, you can squat.
No, I'm just gonna keep running. I'm talking long is going?
Speed strats, both buttons. Just hold both of them down. Oh yeah, it was Dr. Rachel Yohei. She did particle accelerators, and she does research at CERN. So hold both buttons, you'll shoot, and just turn back, and you can shoot. Yep, there you go.
They gave up on me.
It was Dr. Rachel Yohei, you're right.
So that's a tension between, there are so many researchers. So I said earlier that how am I even going to try to get into this car?
So if you go down.
Further down?
So jump in the car, there you go. And if you ride down lower, down, yep. You're going to run into bombs eventually.
Okay.
But if you slow down, maybe up a little, yep.
Okay, fine.
Yep.
Fine. Just keep walking. Just like walking.
But a tension between individual and
funding
consortium.
individuals and funding a few individuals with a lot of money. So we already talked earlier about how the average age of a researcher when they get their first R01 is 44, which, when did you get your faculty position? How old were you?
Well, I was 30. Yep.
So somehow you have to find a way
14 years, yes.
to fund your research, and you have to pay people in your lab to do the research. Should I even bother getting in this? Like, I can't even...
No, this one you want to be in, because if you ride at the bottom of the screen, you'll be good till the end of the level.
Okay.
Just, stay at the bottom.
So we want to give as many people funding as we can. But there are... I can't get over this.
Tap, tap, tap.
Oh,
you have like 80 milliseconds of tap to get over that.
So there are some things that you can only do, though, with a more
Big dollars.
coordinated effort. Wow, that guy is scary. He's making me not want to do drugs.
His name is Kinky Pinky.
Okay.
Because, the classic game member, the clown with a knife.
You know what? He just killed me. I actually might have played this before.
Holy cow.
And the thing that made me, why am I even engaging with you? I don't need to. I'll
Go to the last door. No.
Oh,
Yep, that's fine. It's just dogs.
It's not fine because it's dogs and I hate dogs. Oh my God.
So hold both buttons and turn back. There you go. You turn them into puppies.
Oh.
So go through that door.
Where?
There.
Okay.
Don't.
Can I go through that door?
Nope, you gotta go to the far right. But after you go through the door, stop pressing up.
No, this is not gonna help me get on the board.
Oh, you'll be on the board no matter what.
This is not helping. Okay.
All right, go right. Farthest right door.
Okay. Is that one?
Whoa. What does adult books mean? College textbooks? No, Go Haku. I don't think that's...
Farthest right.
So you might have played this one in the arcade. So this was an arcade big game before it went to the NES. And in the arcade game, they had hookers on this level.
So that's what I remembered is the women wearing...
Yep. No, that was definitely the arcade.
Heels or something.
Yep.
I saw the same.
Yeah, that was this level.
Those people. Like, what are they wearing?
And with NES's or Nintendo's squeaky clean image, they said, no, we're not going to have any mention of drugs or hookers on the screen.
My thumb hurts. Okay.
Worth it. It's worth the suffering.
Oh my God.
It's easy to forget how crippling these old controllers were.
They really are.
Like, they didn't even know ergonomics existed back in the day.
And I can't be at the end, even close to the end, so I've got a lot of thumb pain ahead of me.
Yeah. No, but those aren't cannabis sleeves, because they added extra leaves on them, because you can't mention drugs. This is the only level you go left.
Oh.
And what you're going to have to do is bullet the Rambo guys when they're relatively close, and one of them will drop a card. Oof. Oh, also, some of the marijuana plants are booby trapped.
Those are marijuana plants? They look like ficuses.
In the arcade game, they are. In the Nintendo version, obviously, they can't promote drugs.
Why do I keep jumping when I hit them?
They're exploding you and you jump up.
So, marijuana plants often do.
It's a tale as old as time. All right, go left. Yep. So you're going to bullet them at close range. Go a little higher so you're not at the bottom of the screen. There you go, green card.
Okay.
Now there's a door on the end. There you go. This must be a Home Depot or Lowe's gardening section.
Okay, this is the end of my wine.
Oh, no, that's it. We did it.
I did.
We'll count that as a victory. So yeah, walk right. Man. All right. Gohaku had a question. Has anyone read Code Breakers by Walter Isaacson? I found it hard to read, but want to learn more about CRISPR. I mean,
I have not.
CRISPR is going to change well, it is changing everything, right? It's... How optimistic are you about a future of genetic engineering or like CRISPR for genetic diseases?
Well, I mean, I would like to be optimistic. I think that it has a lot of promise. Ooh.
That car.
The trouble with... Do I have to go to the left?
Go right,
here?
right. Yep. The rest of the levels go right.
All right, sorry. The trouble with these new technologies and getting them into some sort of clinical practice is that you have to manage all of the potential off-target effects that they could have. And you have to really test them well before you... Now, are you going to tell me which door I need to go in?
Yeah, far right.
Okay.
Yep, all these are go to the end. Yep, so
Great.
key card, you already have
Oh,
it.
I do?
You already have it, yep. You have this one and the next one.
How about that?
I mean, CJ, people don't like GMOs. I doubt they'll be into GMing themselves.
I think that when you are very sick, you
will do anything.
You will.
Yeah. Or your kids are sick, or you can give your kids an advantage.
I want to kill that dog. I just killed the dog.
You turned him into a puppy, and he ran away.
These guys look like monks.
Yep. Why are these monks so angry is the classic quote from Ask Scientist Gaming.
What? Like,
Drossokilla, do you stream only on days when you have guests or other days as well? No, typically I only do when I have guests. I've thought about like doing some of my speed runs on off days, but generally we stick to every other Wednesday.
Do I have this one?
Yep, you have all those cards.
Oh. How did I get them? Did I kill people earlier?
They're the ones you got earlier.
Oh, okay.
Didn't Will Smith's character genetically modify the zombies in I Am Legend?
I did not see that.
I think he was trying to study the zombies. And that movie was tragic departure from the book. The book was absolutely amazing, and it completely bastardized the title and the end. If you guys haven't read the I Am Legend book, I strongly recommend you do. Or watch the original Charlton Heston movie, because that's much closer to the book. Because the original ending of the Will Smith movie is actually a better ending than the one they showed.
I actually have no idea what it's about. Will you tell me?
So he's the last person on Earth, but he was like a biologist. So he was taking these zombies and trying to understand them. So wheelchair big, you have to rocket him.
Oh.
Sorry, I didn't tell you were going to rocket a wheelchair guy. But
wow. Seems really counter.
No, but he's a drug lord.
Drugs. Okay.
So,
wait, but how do I?
So tapping A.
I'm not great at tapping.
Yeah, no, this is... So the arcade had 4 buttons. The Nintendo controller only has 2. So you really didn't have an option. Oh, it was his... So CJ says his team made a vaccine for cancer or something and it turned people into vampires.
Oh,
so he actually caused it. I didn't know that.
I am tapping.
Yep,
I'm tapping you.
Oh man, here's the worst part. If you tap on 6 frames, it doesn't fire anything at all.
Ooh,
nice. There it is. Now inchworm. Now you have to do that twice more.
Are you serious?
Yes.
Okay.
Better or worse than Top Gun? You really gotta gradiate your Nintendo games, because there were a lot of shitty ones. We could bust out Bible Adventures if you want. That's a B-rate.
I love how when you kill them, like they just explode in a hail of limbs.
It was really controversial when this first came out. This was one of the earliest where like parents were protesting against games because of that violence. The blood and the torsos and the... Nice. Inchworm.
I just want to get out of here. Look, dog.
Yeah.
Leave me alone.
Also, I would love to do this once a week, but it is such a non-trivial amount of time to set up guests and organize, especially like predictions and stuff. Because like Lisa has never done this before and I have no idea coming into it. I guess you saw a few streams.
I did.
But it's yeah. I
Wait.
would love to do a guest every week. So gold card?
Yeah.
No, exit. All right. We are on to the last boss. So this one
Wait, really?
You're gonna have to go to the top of the screen and then jump rocket him in the hat for the first time. And then I'll tell you what to do.
Mr. Big
Yep. So
lives.
you're gonna have to tap B and then on the way down tap A, but go to the top of the screen and do it.
What is this thing? This is like Java. Wait.
That's Mr. Big's mechanical giant head.
What is he like vomiting?
It is his tongue.
Wait.
So run to the right.
I can't get out of his face.
All right, so run to the right. When you get far enough away, turn around, jump, and rocket him. So tap B and then tap A while you're in the air. So just one tap of B and then one tap of A. No, this is hard. This is not a trivial. Oh. If it makes you feel any better, the asterisk next to Sarah's name was due to me doing this part for her. So if you want an asterisk,
No.
I can help. But go to the top. Go all the way to the top. Yep. So you can
I'm earning this.
All right. So go the other way. Go to the top of the screen.
I'm trying to get away from his face.
Top of the screen. Top of the screen.
I know. I can't. I'm trying to get out. I'm trying to get.
Get a good
further away
distance.
from him.
Turn around. Tap B. Oh, that was close. And then go run away. Try it again. We got this. That was really close. Little less time tapping A.
This is really disturbing. It's just like his face is falling apart and chasing me.
So on our YouTube channel, we have a compilation of our guests playing this particular boss for exact reasons you'd expect. All right, we're in the last 10 minutes or so. Ask a Scientist Gaming. If anyone has questions for Dr. Lisa Chadwick, she is an NIH program director who knows a lot about genetics, genomics,
And
NIH
she's
funding.
way too busy feeding
focusing
this
on
dissolving face.
I mean, you can make her take like 7 drinks right now.
I don't have
Actually, no,
anything
your
else to
wine's
drink.
gone.
Yeah, you're gonna have to
I
give
can
me something
bust out
else.
this- we have things. Tequila, does that hurt? Does that?
I'm not going there now.
Don Carr decided to drink tequila all night.
Oh, don't.
Just straight tequila.
No.
So you could just completely run to the right and practice it. So go around them.
Wait for me.
Fine. Your iframes, go. Go up.
I would drink something else, except I don't have enough time. I'm very busy.
Whips. Oh, there it is. So it's on the downswing. Only
How
takes
many times do
one.
I have to hit him?
So you have to hit him once in the hat.
Oh.
That's it. Yeah, so it's a very particular on the downswing. So you're holding a B. That was close. A little bit later.
Now I understand. It's not helping me solve it, but at least I know what I'm trying to do now.
So if you see him do that little squat motion, you've held B too long. So it's like literally just a little tap.
Okay.
His team made a vaccine for cancer and it turned people into vampires. See, that's why we need FDA approval, ladies and
Yeah.
gentlemen. True story.
This is going to take me all night.
So did you have to fight battles like with family members or anything over the vaccine? I mean,
Me, no.
not at all. God.
But I was prepared to, if I needed to.
Yeah. I mean, you had the insight.
Oh my god.
It's like right when you're at the top of the jump is when you
No.
tap A. Yes. Okay, now rocket him in the face.
What?
Now it doesn't matter if you jump at all. You just rocket him.
I
Now
gotta
he's,
get away from him though.
and now
on
he's
top of
shooting
me and I
lasers
can't-
out of his eyes. I don't entirely know how
That's
that works.
better than his face dissolving on me.
I don't know why
Is this killing him?
Yeah, it should be. Maybe try to get a little bit further away. Its hitboxes are weird. How is that not hitting him?
I don't know.
Okay, try running away a little bit and then turn back.
I saw the results later, and I feel like I'm not doing well enough to
go left. Get a good distance. You can jump away from him too.
Can I? But I forgot how to
B tap. Yep. And then A tap to get rockets. Alright, so run really far left. Get away. You can even go to the top. Yep. Turn around, rocket him in the face. No jumps, just...
Wait.
I don't know why he hasn't gone yet.
He's great.
No, it's a terrible game. Why would anyone play it?
Oh my god.
Okay, stay at the top, turn around, rocket. Oh, rocket. How is that not? There we go.
Oh,
okay,
wait.
so now
What is happening now?
Hold A and hit bullets, and just run away and shoot his spine.
That's fine.
So you can't do it from the top. You have to be somewhere in the middle. So like run away, turn around, and shoot.
This is really stressing me out. Okay. But it doesn't look
So
like it's
you
doing
can't do it
anything.
at the top. You have to be somewhere lower.
Okay.
So there's weird rules. So run away, turn around and shoot. Run away, turn around and shoot. Like, this is how you beat it. Believe it or not, this is how I beat it. Maybe get a little more distance from this guy. There we go, 2 down.
Oh god, I have to get past him again.
So if you, on this next one, if you go to the top like line, you see that line on the screen, like between the tiles, down a little bit.
Yeah.
That line, that gets you closer to hitting him. For some reason, this is the sweet spot.
Where I am right now.
Yep. Or somewhere about there, maybe a little higher. Don't worry, in about a week, I will release the tutorial. Next time you're on.
Great. Thanks.
Yeah, that's perfect. Just walk away.
Die.
Yeah, number 4, number 3. Now just unload and you should be able to get him.
He's rolling over me.
Turn around. Go a little lower, maybe.
Oh, I see. Oh.
Go right. Holy crap. Unassisted, no asterisks next to Lisa Chadwick's name.
Wait, that's the end?
Yes. Get that? Now we're on to civil forfeiture the level. If anyone has suggestions on people we should raid, please feel free to throw those in chat right now. Yeah, now you get to steal gold without any due process.
Great.
Oh, man. Is it everything you hoped it would be?
I mean, I don't know when my thumb is going to recover from this.
23 minutes and 22 seconds.
Okay. I need to know where
you are
I
on
rank.
you are on the scoreboard. 23-22. That's an excellent time.
Ooh,
That
this
puts
is
you at
good.
like 7th. Beat Sarah with no
beat
asterisk.
Sarah, yeah.
That's it. So now you can put in your initials or put in USA or ass. It's up to you. Classics. All right. If anyone has suggestions on people we should raid, you guys should put those in chat right now. But yeah, you only get 3 characters.
Oh.
Count it.
Oh. Pressing the wrong. This is not even going to get saved, but I need to do it.
No, it's a moral win.
Yeah, it really is.
Congratulations.
Thank you.
Like, we didn't beat Top Gun, but we beat NARC and won the war on drugs.
Yep.
Speaking of big funding, if we put the war on drugs money towards research, what would happen?
That would be great.
I don't know.
Drugs
All
are
right.
also bad, though.
No, that's true. Just say no, as the sign says. All right, everyone. Well, that is Ask a Scientist Gaming. You beat that way faster than I expected. We're actually 2 minutes early.
Oh, great.
So thank you all for joining us. It's always a pleasure. The questions really bring us down rabbit holes we're excited to discuss, and the gaming is always an awesome time to relive some of our childhood favorites. So thank you again for joining us. Lisa, any parting words for the audience?
No, I'm really glad that people were here, and I hope that you learned something about genomics and about getting grant funding at the NIH. And if you are thinking about applying for grants at the NIH, my major piece of advice for you is to reach out to a program officer. As you can see, we're normal people, and we're
Exactly.
here to help you. So let us do that.
And listen more than you talk when you talk to them. Take their advice. All right. Again, thank you guys all for joining us. In 2 weeks, we'll have Brad Johnson on. He does geography as well as meteorology, climate change, and the climate change on impacts and in terms of urban climate change. So that should be really interesting. He crosses geography and climatology, which is pretty fun. So that'll be in 2 weeks again. Wednesday, 2 weeks, 8 to 11 as usual. But yeah, thank you as always for joining us at Ask a Scientist Gaming. We have really a lot of fun with this, and your questions really make the night for us. So we'll be rating someone. I'll figure out who that is in a second, but stick around. Thank you as always. We'll see you in 2 weeks.